Hello, my gentle and of course very modern apes. My name is Erica, or Gutsick and this is my channel. And what we do here is we typically investigate topics like biological anthropology, primatology, zoology, paleontology, all that good stuff. And sometimes we do it, more frequently than I would care to admit, through the lens of debunking pseudoscience. So last week I had an insanely successful fundraiser that was only possible thanks to the viewers and supporters of this channel. I was raising money for field work that I'm going to be leaving to go on tomorrow. And when you're seeing this, I'm probably in the middle of Kenya, specifically on the coast of Lake Victoria, looking for Miocene ape teeth, which is just the coolest thing that's ever happened to me, maybe. Anyways, one of the activities, in fact, the primary activity, since it took 3 hours, that we did during this streamathon that occurred last week was we investigated a video, an interview with a one Jeffrey Tomkins, the subject of one of my most recent systematic debunks, this video. And Jeffrey Tomkins kind of responds to my video. And I thought what I would do, since I'm looking for content for while I'm gone, is repackage it for those of you who maybe missed the stream so that you can see it, because I think it's really something. So here is like the approximately 3-hour long clip from that streamathon where we just go through his entire interview with a person who basically watched my video and was trying to show that my video had no grounds, that Jeffrey Tompkins still had a leg to stand on during this interview. So the interview, of course, occurred after the release of my video, and it involves a lot of questions about my video, but because it was a streamathon, we just go through the whole thing, including his entire presentation. So this is, I've got like bite-sized busts on this channel. This is like a Thanksgiving feast. So without further ado, enjoy. Okay, there we go. Can everybody see this? I'm sure you guys all can. I'm sure it's not even a problem. Okay. And then while we do this, I'm going to be figuring out in the meantime how to actually find the rest of the super chats and mark them. Well, actually, I already know how, thanks to Ugly German Truths. Okay, so let me figure out the volume here, and we'll make sure that you guys can hear this. I also need to drink a lot of water. I got to hydrate. I feel like my tongue is the Sahara over here. Okay, I'm going to go to the beginning. And so, okay. Little bit of background here, you guys. This gentleman on the screen here is Apologetics 101. Apologetics 101 was around during my Tompkins video from some time ago, a couple days ago. And Apologetics 101 was like, what a cool, I don't know that I buy this work, whatever, but I'm going to be interviewing Jeffrey Tompkins soon. So like, I'll just ask him some of these questions, and we'll see where we get. And I was very excited for this. So we went back and forth for a little while, and eventually this video came out. So let's hear what our pal here, I believe his name is Brian, has to say. And I've informed Brian that we are going to be watching this video. So I said in the comments somewhere down here. No, wait, that's just me. Okay, sorry, I was on the wrong tab. Here we go. So I said in the comments down here, was my comment deleted? Okay. Not if that's big if true, Apologetics 101. That better not be true.
Hello.
Better not have deleted my comment. Okay, he didn't. Okay, I didn't think so, but someone in the comments said sus, and I was like, maybe. So I said I'll be covering this live today. Looking forward to what Tompkins has to say. So you guys, we're just going to see what Tompkins has in store for us in this wonderful video. And you want to know the best part? Jeffrey Tompkins actually gives a nice little talk in this, and he talks about some paleontology. He goes out on a limb and starts talking about Lucy. And let's see if anybody in the side chat can take a wild guess at what you think Jeffrey Tompkins thinks Lucy's method of locomotion is, because I'll give you a hint, it's not bipedality. Let's get started with this. You guys let me know if you can't hear this.
Hello and welcome to Apologetics 101. I am your host, Brian Keith Bowen. All right, we've of course been doing the Genesis series lately, and I end up interviewing. I have a special interview today. I have an interview with Dr. Jeffrey Tompkins, who's a geneticist from Institute for Creation Research. and it was such an honor to have him on my channel. And we had a great discussion on his method on because he came up with an 84% number as far as chimp and human relationship. He discovered that based on the papers and stuff like that they wrote, that they were cherry picking the data a lot, and so a lot of evolutionists, and so he basically did his own evaluation of it and came up with a technique to eliminate the cherry picking.
Okay, you guys, so we've got a nice little summary of Jeffrey Tompkins's work here at the beginning. What I will tell you is that you probably already know this, but if you're in the chat here, but like, let me fill you in a little bit. Jeffrey Tompkins is a young Earth creationist, and he's a geneticist. He got his PhD from Clemson in, I think, 1996. And he's known for popularizing this number for how similar humans and chimps are to one another of like somewhere between 70% and 89%. Typically, he hovers around 84%, especially as of late. So my most recent video, or one of my most recent videos, what we did is we just tested Jeffrey Tompkins's work. And this was a video that I had been working on for quite some time because it took me months to run all the tests. So with that in mind, I showed sort of in my video through my methodology, which was just copying Jeff's. And I showed that using Jeffrey Tomkins's methodology, whether you're using his 2011, his 2013, his 2015, his 2018, or his 2018, so 2016 or 2018, doesn't matter which one you're using, you cannot get humans and chimpanzees to be 84% different from one another without A, unweighting everything, or B, using ungapped methodology, both of which are highly inappropriate as methods to determine similarity. So let's continue here.
So that it will come up with a more accurate number. And in recent times, this has really been attacked by a lot of critics. When I entered this interview, I didn't know what I was going to find. I didn't know. It's hard to really say with an interviewee because you don't know if they're going to tell you something that you agree with, if they're going to tell you something you don't agree with, if they're going to challenge your assumptions, if they're going to challenge other people's assumptions, if they're going to agree with mainstream views or go against those mainstream views. You don't really know what you're going to find until you actually get there and actually have a conversation with them. A lot of times I'm able to predict what they're going to say because I see what they write in other things, but in other cases, you don't know. So I had no idea. I didn't know whether, I'm not a geneticist. A lot of the critics that was badgering me are not geneticists.
Guys, I don't know if you know this, but I'm not a geneticist. And this is something that I get dogged on all the time for when I'm tangling with young Earth creationists. If you're not a geneticist, you're actually just not allowed to talk about genetics, according to a lot of these young Earth creationists. And to me, this is super hysterical because they do nothing but talk about things outside of their own area of expertise. In this exact video, Tompkins spends time talking about paleoanthropology. He's not a paleoanthropologist, right? But I mean, when he does it, it's fine because he agrees with the YC. So he's allowed to talk out of his area of expertise, but nobody else is. Now, what I'll tell you is that I do think that we have to give kind of a little bit of authority isn't really the word. Trusting experts is important, right? But at the same time, anybody can test methods for themselves, right? Anybody can go out there and investigate certain claims. And, ideally, if you're an expert in a subject, your work should be able to stand up to the scrutiny of non-experts. That's part of the problem with Tompkins, is that I'm not a geneticist, and I was able to show that his work was garbage. But I digress. Let's continue because we've got quite a bit to get through. Got quite a bit.
The criticisms about Tompkins' work is coming from people that are not geneticists, and so here was my chance to be able to talk to a real geneticist about this very discussion. And so, I didn't know what going in. I didn't know what I was going to find. I didn't know if I was going to be proven right, proven wrong, if there was a flaw there, if there's a flaw here. I didn't know what I was going to find. And that's typical of a lot of interviews. You don't really know that much about, what's going to happen. Or I knew that there was critics who had issues with his methodology.
You are done, Tompkins. This method, it's through. It's finished. Throw the towel in.
I knew that there was some young Earth creationists even had.
They put me in there. Sweet. Yes, I will let you know that I do have a problem with the genetics of Jeffrey Tompkins. I stand guilty as charged as a hater and as a critic. But Jeffrey Tompkins did something amazing, right? He posted all his methodologies online for all his different papers, and some of the methods differed between the papers. So, as someone who had a problem with his methods, I was able to check them for myself using controls to see how they fared. And if you'll remember from that video, using Jeffrey Tompkins' methods from 2011, 2013, and 2015, humans are not 100% similar to themselves. Not a population of one population of humans to a population of different humans. But a human reference genome to itself is not 100% identical using those early Tompkins methods. And using the later Tompkins methods, he doesn't weight his data. So a sequence that's 300 base pairs long and 50% similar, right? If you're comparing humans and chimps, let's say, that's going to be weighted exactly the same as a sequence that is 300,000 base pairs long and 99% similar. Both of these are super duper actual flaws in methodology. Not an issue in philosophy, not an issue in sort of like a strategy, right? Not like different ways of measuring. Like they are categorically incorrect ways to compare genomes or to just do math. So we've got a couple of different issues going on here. Oh yeah, a good point, Zach B. Hancock. I checked my work with actual geneticists anyways. That is something that I did do. I mentioned this in the video, right? Like I checked with a lot of experts because I'm not an actual geneticist, right? I just took genetics in undergrad and in my graduate programs. But like, I guess I'm not an actual geneticist, so I'm not allowed to double check Tompkins's work. That being said, eventually in here we're going to get to Tompkins's debut. So let's continue onward.
Questions about how his methodology would work under certain conditions.
No, it's just I guess we could just go back to the fact that it would be nice if Tompkins would reply more, so we could get to the bottom of like what he's working on. He's very hard to contact.
And
oh my god, oh my god, oh no! Apologetics 101 has put me and Donnie on the same team. We're both the bad guys together on this. Me and Team Standing for Truth we're all haters together. the evil cabal. That's that's kind of insulting, actually.
And so I didn't know much of anything going into this discussion, but I did know at least two things. One, everyone wanted answers, and two, we're starting right now. Well, hello, and welcome to Apologetics.
Hello.
I am your host, Christian apologist, Brian T. Bowen. I have here today a geneticist from Institute for Creation Research, Dr. Jeffrey Tompkins. We're gonna go ahead and jump in. Dr. Tompkins, if you want to go ahead and introduce yourself and then show your I know you have a PowerPoint presentation. I have some questions here, but I'm gonna save those until after you get done with your presentation. You might even answer a few of them. So take it away.
Okay, so I feel like we don't need to go through Jeffrey Tompkins' introduction. I've watched a lot of Jeffrey Tompkins videos, and I've seen his introduction several times. He basically says the same thing over and over again. Where he got his undergraduate degree, where he got his genetics PhD, it was at Clemson, some of the undergraduate work that he did with plants. And critically, something that he mentions in almost all of his introductions is that he was like brought on board by ICR to investigate the human-chimp similarity claim, and that he went in without any presuppositions. He says he's going in without any ideas of what the similarity would turn out to be, which is something, of course, that we all definitely believe him on as a young Earth creationist. So I'm going to skip past his introduction here, and hopefully we're going to get to his presentation on created kinds. So guys, we're going to kind of zoom through the presentation. Like I've done a little bit of investigation into this video thus far, and like eventually right around here, this section here at about an hour and 6 minutes is where we get to the stuff where Tompkins is actually trying to answer questions from me. Otherwise, he spends a lot of time talking about created kinds, and he spends a lot of time talking about genetic entropy. He talks about how devolution is going on. I'm tempted though to go over some of the presentations. Specifically, I'm tempted to go over evolution in Australopithecus because, as some of you guessed in the side chat, Tomkins does think Lucy is a knuckle walker, which is not true. So I'm gonna kind of put the feelers out. I'm gonna see what are you guys saying over there? What do you want? You think we go through this whole thing? You think we go through this whole bad boy right here, or you think maybe we skip to the questions? What sounds more fun to you? Whole thing or not whole thing? What do we think? You guys let me know. Okay, James Hampton is the only chat that I see, and he's saying do it. All right. Okay, I'm getting a lot of whole things here. Yeah, I'm kind of doing a poll, Brie, kind of. Okay, now we're getting a whole, okay. All right, you guys. All right. We want to do this whole thing? I'll do the whole thing just for you. We'll check out the whole full Monty here. Savage Crow was like, I don't have anything else to do today. Me either. All right.
My PowerPoint okay?
Yeah, it's okay. I like your monkey.
So I call this monkey business in the chimp genome, and you'll basically see what I mean by this as we get going. And now my slides aren't advancing for some reason. No. There we go. Okay. So how did this whole idea of humans evolving from an ape get going to begin with? Well, really, Charles Darwin was the big impetus for this in the late 1800s. He proposed that humans evolved from apes. And of course, Darwin got this whole idea of a tree to Darwin. Life evolved from of course, Darwin didn't have all the biology and genetics that we have now, but basically creatures evolved over deep time and from a single-celled organism originally that somehow miraculously came into existence and then created the diversity of life that we see now in this tree-shaped pattern. We call it a phylogeny in the genetics business, but that's really not what we see in the real world. We see what myself and a number of other creation scientists would call a creation orchard. So we see lots of different kinds of creatures, plants, and animals, and we see variation within those kinds of creatures, but we don't really see one fundamental kind of creature or type of creature evolving into another one.
Okay, so, oh God, we're going to be here all day. We're going to be here forever. This is going to last years. This whole thing, every second. Oh my God. All right. Okay. Let's back up a little bit. So yes, Darwin was one of the first individuals to propose that humans descend from some kind of precursor ape, but we also still are apes, right? Humans are still apes. We have all of the characteristics physically and genetically that designate an ape or a hominid as such. We also have the hominoid traits and the catarrhine traits and the anthropoid traits and the haplorhine traits and the primate traits and the mammal traits and so on and so forth. We have them all, and if you were to remove humans, right, and say, let's categorize all life except for humans, you would end up with the same categories that we have today, right? You would end up with primates as an order, and you would end up with carnivorans, and you would end up with all sorts of like Felidae and Canidae as families, and Hominidae, and blah, 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 blah. And then let's say you come back and you say, now that we've established this tree of life, where would humans fit in potentially? And we would fall within class Mammalia, order primates. We'd fall into the haplorhines and the anthropoids, and we'd fall into the catarrhines and the hominoids and the hominids and hominins based on physical characteristics. And then also we would nest that way with our genomes as well. This is not arguable, right? For this reason, Ken Ham had to tweet the other day that he thinks humans should be in a different kingdom of life on the basis of the sole parameter created in the image of God, right? Like you cannot do that. That's not something that is standardized or consistent or even biologically real. It's just something Ken made up and thought would be cool to post on Twitter, I guess. Now, secondarily speaking, here discussing the difference between an evolutionary tree and a creation orchard, where's the line, right? Where do you draw the line in these different kinds of organisms? They can't draw a line morphologically speaking. They can't draw a line genetically speaking. Right? Which means they can't even, it's not even just that they can't draw kinds in extant organisms. They really can't draw them when you take the fossil record into account, right? Like, are amphicyonids a different kind from ursids and from canids? Like, because you got bears, you've got dogs, you've got dog bears. Where does the line fall there? are Australopiths their own kind, right? Because some guys, like it Answers in Genesis, think that all of the apes belong into a kind, and it's like Pongidae, right? And then you have humans as being something separate. But I know for a fact that Tomkins believes that chimps and bonobos are a kind, gorillas are a kind, and orangutans are a kind. There is absolutely no standardized method for anything with regard to baraminology and kinds. It is patently absurd and not applicable at all. But I mean, I think the reason they do it is because it feels intuitive. Like they sit back and they're like, look at all of these different groups of organisms. Everybody comes up with categories for organisms. We're humans. We like to put things into bins. And then they're like, this thing that feels intuitive after having undergone numerous different iterations through time, especially after the advent of genetics, this is actually just more or less the created groups that God made, except when it isn't. Like, there are people in the chat who are like, oh, kind is at the family level, kind is at the genus level, kind is at the order level. And like, I'll tell you, it's all of those things. It's whatever they need it to be at that given moment in time. Like, for a video that I'm working on right now on the fusion of human chromosome 2, I did some research on on chromosomal fusions. And chromosomal fusions occur in suids, right? So in members of the pig family, so pigs and babiruses and warthogs and whatever. And like the Ark Encounter thinks that these are not a created kind. They actually accept Suoidea, right? Which is the superfamily of pigs. Why is it at the family level for dogs and at the family level for cats, but at the superfamily level for pigs? There's no methodology for any of this, you guys. And that's what's so frustrating. It's not just that the methods aren't standard, right? They aren't, but they're not even like internally consistent. So it's incredibly frustrating. But let's let old Jeff here get on with it.
And so here's just kind of a visual. So we see humans all over the earth, and there's a lot of variation among humans. And then dogs and other kinds of creatures. There's roses, many different forms of roses and things, but we never see anything evolving into really anything else. Just
okay, so you wouldn't, God, like Jeff is a geneticist. Like he should know some of this stuff, right? The law of monophyly is like a standard that we use when we're talking about the evolution of organisms through a given lineage. Nothing is going to become anything else. They further specify. Right? Nothing is going to transform into a different family. Nothing's going to transform into a different order or into a different species. They're going to further specify down through the tree of life. That's how this works. But please appreciate here that we have on the top several different drawings of people, and then below we have different dog breeds, and then below that different species of frogs. So these are just categorically already 3 separate things. All of these are kinds, but they're separate things, right? So like this is a kind, and these are all just humans. This is a kind, and these are all different breeds. These are a kind, and these are all different species. And then I don't know enough about roses to tell you anything about these roses down here. But like already right off the bat, in the picture that he's using in his own tailor-made presentation, the kinds are inconsistent, and it is not going to get better. Prediction.
a lot of variation within different kinds of creatures. So how did this whole idea in the modern era, anyways, of chimpanzees becoming the closest ancestor to humans get going? Well, we'll talk about that and what that means here as we move along. But I think it's safe to say that most modern scientists would claim that the chimpanzees are our closest living relative. And they largely do this based on the DNA sequence that they claim is more similar to human than some of the other great apes. And so a standard evolutionary tree that most modern evolutionists would claim would have humans up there at the top of the tree, and then chimpanzees, and then they would say we're more closely related after that, the gorillas, orangutans, and then there's rhesus macaque, a monkey, as an outgroup there at the bottom. So that would be the standard phylogenetic tree for humans and their so-called living ancestors. But evolutionists can never really agree among themselves on most things. They always have debates. And so this was an interesting paper.
I'm sorry, living ancestors? Living ancestors. I've never once heard that as a phrase. And I'm a biological anthropology student. They're closest living relatives, but they're like cousins, right? They're no more a living ancestor than I don't know, than like a fox is to a maned wolf, right? We're related to them. They're some of our closest living relatives. not living ancestors. That applies like an ancestor-descendant relationship. Like they have changed not at all since we've split from a last common ancestor, which is, by the way, you guys, patently false. Chimpanzees have been evolving for just as long as humans have. We've both been evolving for the same amount of time, and they've changed in many ways that we haven't changed, and of course, vice versa. So my favorite example for something that humans are still pretty primitive in, although it's an acceptation, is we have really long thumbs. So our thumbs are super long compared to the rest of our fingers. Chimps have really long phalanges and really long metacarpals and short stubby thumbs. We have really long thumbs and stubbier fingers. And this is because we need to be able to maintain contact with all of our fingers. This is a characteristic that existed in proconsul and akembo, right? This is characteristic of, more characteristic of like a palmagrade, pronagrade, arboreal quadruped than it is of a knuckle walker. And we simply took that and we were like, why change it? Let's just accept it and use it as sort of a jumping off point for a precision grip. And yes, someone who commented in the side, he did say macaque, which is something that I've not heard as a pronunciation, which is kind of funny. Okay.
It's fairly recent, 2009. And actually the authors of this paper claim that humans are more similar to orangutans than they are to chimps. And they did this based on the morphology or the anatomical characteristics that humans share with the various great apes.
Yeah, so Jeff Schwartz, who's the second author of this paper down here, is notorious for this opinion in the paleoanthropological community. He thinks that humans superficially share more morphologically with orangutans than we do with chimpanzees. And in some ways, he is correct in some ways. For example, certain aspects of our knees are going to be more similar to orangs than they are to chimps because the similarities, morphologically speaking, necessary for suspensory locomotion and bipedal locomotion are more similar than those for terrestrial knuckle walking. So like, yeah, there's going to be some things that we share more with orangutans, but this is something that they don't understand. And I can't for the life of me wrap my brain around it. It is the preponderance of characteristics that is the best for determining like who your closest relative is, whether that's morphologically speaking or genetically speaking. And I'll show you what I mean here in a second.
And so there isn't a complete consensus on this, but I would say based on the DNA, yeah, humans probably are more similar in their genome to chimpanzees. But the fact of the matter is that chimps, orangutans, humans, and gorillas, they're all just separate kinds of creatures. And they all have their unique similarities. And so humans kind of share, it's the mosaic. We share some traits with orangutans and some other traits with gorillas and some with chimps. So it's not really a clear-cut deal by any means.
Okay, so pause here. It is clear cut, right? So that paper was from 2009. He's like, yeah, this paper's pretty recent. It's just from almost 2 decades ago. Like this is also comparative genomics and paleoanthropology, biological anthropology. So this is not exactly a slow-moving field, series of fields, right? But that being said, this is not controversial to say that, okay, so humans and panins, humans, chimps, and bonobos, we share the most with one another, morphologically and genetically. Then us as a group of 3, us as a trio, secondarily share the most with gorillas. And then us as a quad, then sequentially share the most with orangutans. And then us as sort of a fivesome, then share the most with gibbons, right? So this is a nested hierarchy, genetically and morphologically speaking. And this is when we're looking at, surprise, the whole genome, not 1 or 2 traits, which you can already see what he's doing here. This guy's a geneticist. I don't understand. He's like, did you know if you take a certain protein or a certain gene or this, that, or the other thing, whatever, a certain sequence even in the genome, sometimes you're going to get a different phylogeny? It's like, yeah, you will sometimes get a different phylogeny, right? Like evolution and the way our genomes evolve is not so clear cut as to eliminate things like incomplete lineage sorting. So with this in mind, there are going to be some minute things that we share more with gorillas or that we share more with even orangutans, things that we've perhaps both retained from a common ancestor that have maybe changed in gorillas or in chimpanzees. And as a result, if you create a phylogeny on that one trait, you're going to get a different phylogeny. What if you use the whole genome, though, you guys, which is typically what we do, and why every single comparative genomics person on the planet agrees that humans and chimpanzees and bonobos, so panins, are one another's closest living relatives? Because overall, on the whole, when you consider every gene, we share more with them and they with us than either of us do with anything else. It is the preponderance of characteristics. But like, why is he not appreciating it? It doesn't make any sense.
And in fact, the first thing that I discovered was this mosaic type of situation when I began investigating the molecular basis for human similarity with great apes. And so this is just a protein sequence, and proteins are chains of amino acids, and proteins are the workhorses in the cell. And so this is a protein called alpha motif protein 11, and I ran this in software that created a phylogenetic tree. And I did not get the standard evolutionary tree for this. In fact, gorillas for this particular protein were more similar to humans.
Okay, guys, like we just want to, I'm going to skip some of this. I hope you guys don't mind. We just went over why you don't create phylogenies over a single gene, right? Like there are minute differences between me and my sister that are going to be different between the two of us that maybe I or her retain from our grandparent and share with a cousin. And that's going to create a different phylogenetic tree if you just use that one gene than if you use the full genomes, which of course show me and my sister to be most similar to one another. Okay, so he uses the alpha motif protein, ACAP3. Okay, that seems to be the end of that.
And that's, like I said, that's because each kind of ape and humans they're their own created kinds of creatures. So when I began investigating the literature regarding the human chip DNA similarity issue, which is really where you begin in any research project, you have to see what the literature is saying. This was one of the first papers that was published comparing human with a number of ape genomes. And what they did is they took the most similar sequences they could find between humans and great apes. So they cherry picked the data right up front, and then they ran it through software to construct evolutionary trees or phylogenetic trees. And this is a quote from the paper. And so this was a study that was published where they compared the most identical sequences they could find between humans and great apes. So they cherry picked the data right up front. But anyways, they ran the DNA sequences into a software program that constructed evolutionary trees. And they said this in the paper: for about 23% of our genome, we share no immediate genetic ancestry with our closest living relative, the chimpanzee. So in other words, 23% of their evolutionary trees were all mixed up, just like I showed you in the previous slide. So there wasn't any clear pattern of human evolution from apes. It was a mosaic type situation.
Okay, so here's the thing with that though, right? Like, again, I'm glad we're looking at a super recent paper from 2007-1, but yes, incomplete lineage sorting is something that's been appreciated in the great apes for quite some time. We split from panins 7 million years ago, and us and panins split from gorillas about 10 to 13 million years ago. So not that much like further back into the past. And it's been proposed on the basis of genetic work like this, that the ancestors of us and panins, like the shared common ancestor of us and panins, could interbreed with the common ancestor, with the ancestor of gorillas, the gorilla lineage at that time. And that also after humans or hominins and panins split, genes could still be exchanged for like 3 million years. So these genes are exchanging across basically all three lineages for quite some time. And that's where you get incomplete lineage sorting from. Incomplete lineage sorting is not like an unknown factor, right? It's a facet of how organisms diversify and evolve through time. So I don't understand really what the point is here, except that he's trying to cast doubt. He's like, oh, look, they're getting different results. It's like, no, again, I draw your attention back to what we just said previously. When you compare the entire genome, humans and chimpanzees are the closest to one another, right? Chimps aren't closer to gorillas than they are to us. They're the closest to us and us to them and bonobos, so panins in general. And then gorillas are next. And there are some characteristics that we share with gorillas that we don't share with chimps, or that chimps share with gorillas that they don't share with us, right? Like this is just how it works. This is a process that's pretty well understood. But I don't think the purpose of Jeff's presentation here is to actually investigate concepts like incomplete lineage sorting. It's to cast doubt on the process. Fortunately, we were looking at a slightly more recent paper just a mere decade ago in 2012 called Insights into Hominin Evolution from the Gorilla Genome Sequence. And 30% of the genome of the gorilla is closer to the human or the chimpanzee than the latter are to each other. Yes, again, this trifecta is the result of incomplete lineage sorting through time.
In 2012, they sequenced the gorilla genome, and they got the same results when they began constructing these phylogenetic trees with human DNA and the great apes. And they said in 30% of the genome, gorilla is closer to human or chimpanzee than the latter are to each other. So once again, a certain percentage of their evolutionary trees are not fitting the paradigm. They're indicating more of a mosaic sort of situation between the different genomes. Now, having said that, what is the actual DNA similarity between humans and chimpanzees?
Well, Jeff, excellent question. I'll answer that for myself to let everybody know here. It depends on what you're looking at. If you're looking at protein coding regions, the most recent number that I have seen is actually from a comparative of great ape genomes, from 2018, and it's 99.1% for humans and chimps for protein coding regions. The number I always quote is 98.8, but that's actually a little bit out of date. We're even more similar to them now that more work has been done. At 99.1% for coding proteins. Now, that doesn't include indels, so that doesn't include regions of the genomes where large insertions or deletions have taken place. And it also depends on how you measure those indels, right? Do you count an indel as a single event, as a single difference, or do you count every base pair as a difference within it? Now, the conventional number that we get for the full genome is 95 to 96% similar, which again is still more similar than like either of us is to anything else that is alive today. So very close indeed. But Jeff likes to do the 98 to 99 first, so then he can be like, but then they changed their mind and actually it's only 9. Now they're only saying it's 96. It's like, I don't really know that that's a fair characterization of the progress on the field, but I digress. Okay, let's continue here.
Is it 98 to 99% identical? Then I began tackling this issue. And what's interesting is that at the beginning of the chimpanzee genome project, which we're going to talk about in a little bit, they took 3 million sequences, DNA sequences at random from all over the chimp genome, and they compared those to the human genome. And this is a direct quote from the paper. They said about 2/3 of the, in other words, of the chimp DNA sequences, only 2/3 could be unambiguously aligned to DNA sequences in humans. So only.
Okay, so not only 2/3, about 2/3. So you see how we have like a subtle little word swap here. This is also from 2002. So this is from the genome-wide comparison. This is before the chimpanzee genome was actually published. It was published in 2005. So I'm not really sure that this is an accurate slide to be having on the screen right now to be talking about the similarities between humans and chimps when you're not using the most up-to-date version of the human genome or the most up-to-date version of the chimpanzee genome.
Only 2/3 of the chimp DNA, could they even match up accurately onto human? Well, that doesn't sound like a 98 to 99% similarity to me if they can only match up about 2/3 of the sequence.
Well, given, again, this is before the full chimpanzee genome was sequenced, I'm not surprised.
And so I began actually investigating all of the papers where they claimed a 98 to 99% DNA similarity, and there were 6 major papers that were published on that issue. And so I published a paper called Genomic Monkey Business: Estimates of Nearly Identical Human-Chimp DNA Similarity Re-Evaluated Using Omitted Data. So I looked at all these 6 papers, and where I could factor data back in that they had basically thrown out and discarded, I came up with actual similarities of 66 to 87%, depending on the paper. So obviously something was going on here.
Now, this was before, to my understanding, this is before his 2011 paper. So this is before he's actually running any actual tests for himself. This is just him going into the supplementary material of some of these really early papers and coming up with numbers that he feels are more accurate, probably by using, changing how you're actually counting things like indels, or changing how overall similarity is determined by looking at the total number of regions that were capable of being aligned. I don't really know that this is honest either, because like the chimpanzee genome and the human genome are different sizes. So what Jeff, at least in my experience, has done a lot of the time is he's taken the portions that don't actually align, like because they don't exist in one or the other, and he counts those as the maximum amount of differences available. So like, I feel this is not exactly appropriate, because like, okay, let's say I have a play, right? And let's say it's Romeo and Juliet. If I have Romeo and Juliet and I have that in one hand, and then I have a compilation of Shakespeare's plays in the other, right? Romeo and Juliet is 100% identical to a portion of Shakespeare's playbook, because the entirety of Romeo and Juliet is located in that playbook. But the playbook isn't obviously 100% identical to Romeo and Juliet because it has a bunch of additional information. So it depends on how you actually count this stuff. That's important information. And if you'll remember from my Jeffrey Tompkins video, Jeff isn't exactly great about being super clear about how he's actually coming up with these additional numbers. So it's great that he's come up with these numbers here. I haven't looked at this original genomic monkey business paper here because he doesn't, again, do any of his own work in it. But it wouldn't surprise me if he did in that what he's done in many others, which is, to me, kind of fishy. We'll let him continue because I've got a little bit more to say here.
And in fact, what's interesting is that the first draft of the chimp genome, which was published in this 2005 paper, they didn't actually say what was the DNA similarity genome-wide. They just talked about the DNA similarity for the specific regions that they could align and match up. And so that's what's kind of deceiving about this paper, is because those regions with the parameters on the algorithm they had seemed to indicate a very high similarity. But when I actually took the actual alignment similarity, multiplied it by the size of the chimp genome and the human genomes, factored those in, I came up with an 81% similarity maximum from that paper.
Okay, so this is exactly the thing I was just talking about. He has a beef with how comparisons are run with genomes of different sizes. The problem with this is human genomes can also differ slightly in size, and you have to actually have a standardized methodology for dealing with this kind of discrepancy in a way that's not going to, because how you run it in BLAST, like penalize the comparison. Fortunately, since the original sequencing of the chimpanzee genome, we've completed a great more of the chimpanzee genome, and we have the entirety of the human genome. And I have not seen that number of 96% actually change for the full genomes being compared to one another. And as I said, the protein coding number has actually gone up. So I'm going to press play again and also let my dog out of this room because he's scratching at the door. Hold on.
But I had to do that basically going in and working with the data that was there, because they wouldn't admit this up front. So how have evolutionists achieved estimates of high similarity? Well, first of all, they've omitted the non-similar regions of the genome, which in some ways you can't fault them for that, because if the algorithm can't compare regions that are so dissimilar, or you have a region, you're just not going to get any data because there's nothing to compare. And so there are large regions of the chip you just flat out can't match up because they're so different. And the one thing that I did notice was they were only comparing the highly similar segments, particularly the protein coding regions of the genome. They like to focus on those because those did kick out a high similarity.
Okay, so these are two different things, though. So omitting non-similar regions of the genome, or initially this was done because there were like regions of the genome that weren't actually sequenced, so highly repetitive, like TTA, 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 over and over and over again. And that second one compared only highly similar segments. This is not the case today, right? We've compared the genomes to one another like in full, and the number has stayed the same. So like Jeff does this though. He likes to argue with earlier versions, earlier papers, because I guess he thinks he has a better case with those, at least because they were when the analyses were actually less complete. The genomes themselves were actually less complete than they are today.
And so what do we call this? Well, I call it cherry picking the data. So in other words, these high estimates of DNA similarity, 98, 99%, were basically cherry-picked data from only the most similar regions of the human and chimp genomes, with everything else being basically omitted. So let's talk about the human genome real quick, because it's important to keep this in the picture when we talk about how the chimp genome was sequenced, because there's a big difference between the two. So the human genome, the first draft was published in two separate journals, Science and Nature, because there was two separate consortiums. There was a private sector project led by Craig Venter, and then there was the public sector project led by Francis Collins, who was the head of the Human Genome Project at the time. And they agreed to, you can see them shaking hands up there, they agreed to publish their genomes at essentially the same time. And so those were published in 2001, and those were that was the first rough draft of the human genome. And then the chimp genome was published in 2005 as far as the initial sequence.
Okay, so he knows when the chimp genome was published. So why was he using an earlier 2002 publication? Right? Like what's the reasoning behind, I mean, I know what the reasoning is behind some of these decisions here.
In the initial draft of that. So what's the difference? Well, the human genome had billions of dollars of funding. There were labs all over the United States, Japan, Europe, really all over the world that were working on this project, billions of dollars in funding. And so they did a very methodical job of sequencing the human genome, but the chimpanzee genome project was very limited in its funding. And so they did not have near the amount of resources to sequence the chimp genome. And I'll explain why that is important. Well, let's talk about DNA sequencing. What is it? You know, a lot of people, they watch shows like Star Trek and they think, oh, you just stick something in the computer and out pops a genome. Well, it's not quite that
okay,
simple.
Jeff, you're killing me with this.
So let's talk about the human genome sequencing project. It was methodical.
Okay, I'm going to skip through some of this because he's just talking about stuff we've already covered in other videos, like how you have to be specific in how you run comparisons, lest you fall victim to basically bogus results because you're not actually following a consistent methodology. So he's talking about a page-by-page sequencing strategy. We've discussed this. We discussed this earlier in this video here. How do you actually run alignments? How do you align genomes to one another? He's talking about the original, like, I think this is like a shotgun sequence approach that we did with the original chimpanzee genome. And his entire purpose of talking about all of this, as I said in the Tompkins video previously, is because he's arguing that it's humanized. He's arguing that the chimpanzee genome was inaccurately assembled to look more similar to humans than perhaps it should, because the human genome at the time was being used as a reference. And he is correct that there was a lot more money behind the Human Genome Project than there was behind the Chimpanzee Genome Project. I feel like that makes sense. We're a narcissistic species. We do engage in a little bit of zeroing in on our own species. But since then, we have completed the chimpanzee genome without using the human as a reference. It's called Pantro6, and it's the genome I used in my analysis replicating Tompkins's work. So all of that stuff that we just talked about how, you know, the chimpanzee genome is too humanized, it's going to give erroneous results, whatever, that does not matter for the purposes of the analysis that we ran. And again, that analysis showed that Jeffrey Tompkins' methods don't work because they show a human as being like roughly 79 to 87% similar to itself. So let's continue.
A brand new DNA sequencing technology was employed that produced very long DNA sequence reads compared to the old technology. So this new technology would produce DNA snippets that were 30,000 bases long or more, compared to the old technology, which only produced DNA snippets 300 to say 1,000 bases long.
By the way, just in case you're curious, that paper right there, Cronenberg et al., high resolution comparative analysis of grade 8 genomes, that's where I got the new increased number of human chimp similarity for protein coding regions being 99.1%. So like, he knows that paper exists. He knows that we've actually run these comparisons with like non-humanized versions and that the number has not changed. That's like, if memory serves, this sentence that he's got up here is actually, I think it's in the introduction, which is like, that makes sense because they're saying, here's what the previous problem was, here's how we have addressed it.
And so this new technology was very effective in producing a high quality chimpanzee genome. And in fact, in this paper, what's very interesting is that the authors of the paper admitted that previous versions essentially humanized. And so in this project, they did not use, initially anyways, the human genome as a scaffold to align all of the chimp sequences onto, because the sequences were so long, they could basically assemble them on their own merits. And we call this a de novo assembly. And so as what they said in the paper, let me move this little thing here. They said the higher quality human genome assemblies, speaking of the previous ones, have often been used to guide the final stages of non-human genome projects, including the order and orientation of sequence contigs, and perhaps more importantly, the annotation of genes. That's an interesting statement because I actually discovered they were inserting human genes into the chimpanzee genome electronically. They actually admitted this on a Genome Center website.
Okay.
Viewing comment of a
on here.
paper, and he said, so anyways, line them up onto the human genome. So anyways, this is another quote from the...
Guys, what'd the paper say? Right? Like, he doesn't talk about the results of the paper that he just cited. The results of the paper, which show us that the human genome is still, even with these more remarkably complete graded genomes that were assembled using Tompkins' words here on their own merit, the ones that I used in my analysis replicating his methods, it's still 96% for full genomes and 98 plus, 99.1 actually, for protein coding regions. But he can't talk about that part, right? He wants to take the portions from the introduction that are introducing the problem that this paper seeks to solve and then say, you see, they've admitted it. That's the language he uses. They've admitted that there's a problem. Anyways, moving on. And it's like, no, talk about what they actually did, how they improved upon the previous genomes, and what the results were. But he doesn't talk about it. Of course he doesn't.
The same author that was authored that previous paper, and he said in a separate paper, non-human primate genomes have not been finished to the same standard as the human reference genome and typically carry hundreds of thousands of gaps. precisely over these regions of complex variation. So not only did they admit that the ape genomes had been humanized, but that also the quality of the ape genomes was not even close to being the same as human. But there's more problems going on than ape DNA being lined up onto the human genome and human.
Okay, quick note here. Yeah, no shit. It's not going to be as complete as the human genome, because the human genome was worked on like exclusively for a great many years with hundreds of people, if not thousands of people, all working on it. The chimpanzee genome project was considerably smaller, but it is still remarkably complete right now as compared to especially where it was. And certainly it's complete enough to run these comparisons. And I guess Tomkins would say that the comparison, like the portions of the genome that aren't finished, he's just going to chalk that up as being like definitive differences. But like that being said, we'll move on for now. He's going to talk about how human DNA has contaminated non-primate genome databases. Okay, yeah, contamination is going to happen, right? You need to show us that the contamination happened specifically in the analyses that you are criticizing here, right? Like there's things all the time that happen in science that are going to impact a certain area and impact the results of a certain area. But like, if it happens in one place, that doesn't mean it's happened everywhere. And I think that's kind of on him to show support for.
Humanizing these ape genomes. Another problem is human DNA contamination in genomes that are out there in the public databases. And so this paper in 2011 was published, Abundant Human DNA Contamination Identified in Non-Primate Genome Databases. They found
some of this, because again, he's not going to say anything.
That are not human.
Yeah, that's just
DNA full in B.
That's
and the various admissions that are being made about the state and the genome accurately assembled if it's put together based on the human genome. Another question I had is human DNA contamination a problem in the chimpanzee genome?
Okay, so my understanding is that he's asking these questions sort of in reference to, he's asking these questions like saying that these were the ones that he had prior to his 2018 work, I'm assuming. Because the 2018 work, he utilized the new raw chimp contigs that were published, right? The precursor, the preface to Pantro 6, the most recent chimpanzee genome that wasn't assembled using a human scaffold. So I think what he's saying here is like, okay, I was wondering like, what would the results be if I used Pantro 6 instead of previous versions? And also presuming that Pantro 6 is not contaminated, what would be the results if I used a non-contaminated genome? I think that's what's going on here. I hope that's what's going on here, because I know for a fact that he talks about how the Pantro 6 version or at least the raw contigs in his 2018 paper aren't contaminated and are correctly assembled. So I certainly hope he's not forgetting that he said that.
If it was, that would jack up the similarity as well. So what is the actual DNA similarity between humans and chimpanzees, and how can this be determined? So let's do an analogy here and talk about DNA similarity and what we actually mean by that. So here's a theoretical sequence of human DNA and then chimp DNA. And every time you see a vertical bar, that means there's a match. So where there's a T in human, there's a T in chimp, a C in human, a C in chimp, and so on.
Great. Good
stuff.
So
this particular example, there would be a 100% similarity. But let's say you have a C in human at one place and an A in chimp in another. And so that would be a mismatch, a single base mismatch. So in this example, 27 28 bases match, and that would be a 96. One of the problems in DNA similarity as well is not the fact that we have a different base at a certain position in human versus chimp and vice versa, but that you also have these things called insertions and deletions, single base up to perhaps even hundreds to thousands in human compared to the chimp sequence or vice versa. So these insertions and deletions will decrease the similarity. So in this particular example, if we just counted the substitutions, we would have a 98% similarity. If we included the insertions and deletions with the substitutions, we would have an 81%.
Okay, so yes, this is how we get the two differences that we've talked about already, like in this video. If you just use substitutions like your single nucleotide polymorphisms, right, you're going to get a ridiculously high similarity. That's like our 98 plus number. But if you include indels, that's where you get the 95 to 96% similarity. Those are the conventional numbers, right? That's how it hashes out. So Tomkins is right to ask this question, but this question has already been asked in conventional science and has given us two different concrete numbers that are like universally accepted. So him asking this question is basically, in my opinion, him just saying, look, like we have to consider this stuff, and kind of neglecting to point out that it's already been considered. Because the problems that he has in his own methodology, the gapped versus ungapped and the weighted versus unweighted, are actually separate from this. This is a separate issue from those two things. So like, I want to hear him talk about that.
And so sometimes the algorithm can account for smaller insertions and deletions, but when they get too big, then the algorithm can't continue matching. It just breaks off. So I actually did a very large project where I looked at all 101 chimpanzee trace read data sets. And I basically sampled 25,000 DNA sequences at random from each of the 100 software with a random sampling algorithm to do this. But anyways, I was able to create a data set of 2.5 million DNA sequences from chimp, 25,000 from each of the 101 data sets. So on average, these DNA sequences were about 700 bases long, and then I matched them onto human. And so I only recorded what I call the best hit, or the longest match, because you'll get little snippets of DNA in a sequence kind of matching all over the place because of perhaps it's a little repetitive or whatever. So I would only record the best hit. The nice thing about the Sanger style sequences that had a lot of what we call metadata associated with it. So there would be data with it and so forth. So at these, I saw what time Genome Project similarity for these reads.
Okay. The problem with it, we've gone through all of these papers. I went through them meticulously, paper by paper, in my Tompkins video. So I'm not going to ask you guys to sit through him talking about it. I wouldn't do that to you. This that he's got here, this is just patently incorrect in the literature, right? Like the 2002 to 2004, them having a 92% similarity and then going down to 85% in 2005 to 2011. That is patently false, and you can find this out for yourself by doing a literature search. Now, him saying that he's gotten these numbers, I suspect, again, has to do with the weighting versus the unweighting and the gapped versus the ungapped. I know that in his 2016 paper, which isn't included in the brackets here, in 2016, he didn't use any controls, right? So like he didn't publish his CSV to my understanding. So like you can't go through it the same way you can go through it in his 2018 paper. But Josh Swaminathan took his exact parameters, did a much smaller scale comparison, and found that it doesn't pass control tests. A human ends up being 87% to themselves, the reference genome to itself, using the 2016 methods here. So he can talk about all these liberal gapping parameters and what have you, but there's something funky going on in his 2016 paper. And given this is after 2015, I would wager it's a problem with the weighting. But we'll get there in a second.
With human was 92%. For each, on average, 92% was the average DNA sequence identity for these reads. But during the last phase of the Chimp Genome Project, 2005 to 2011, the DNA similarity on average was only 85%. So why the 7% difference? So what's interesting is those years, 2002 to 2004, were actually the years that went into the draft genome of the chimp project that I showed you that was published in 2005. So I think that in the early phases of the Chimp Genome Project, there's a lot of human DNA contamination in the data sets. That's why they were really a whopping 7% more similar to human than the sequences at the latter end of the Chimp Genome Project. Contamination, in my opinion, well, that presents a huge problem to the sequences being lined up onto the human genome.
So I love this, right? Like he's like, by my calculations, the contamination, or by my calculations, the similarity has gone down. So what happened early on? Probably contamination. And he's just asserting that on the basis that contamination has happened in labs before, right? And that it's been polished on. That's what the evidence is. Nothing specifically with relation to the chimpanzee original genomic sequence, nothing like that. Just it's contamination. We'll just call it a day. I don't know how robust that is, Jeff.
It was a scaffold chimp genome sequence that was put out in 2018. It was a new technology. And so I had 18,000 segments at random from around the chimp genome that I lined up onto the human genome. Those DNA sequences were on average about 30,000 bases long. But of those 30,000 base chunks, I could only line up 10,000 bases on average. So in other words, only a third of those 30,000 base chunks of chimp DNA would even align to human on average. And
okay, so this, on the other hand, this you can find. This you can find precisely. So let's just go through it real quick. Let's go to his conpigs, because I've looked this up a gazillion times. So you guys can do this for yourselves. Can you see this? No, you can't see it because I'm on a different thing. Okay, hold on. I've got this. I can handle this. I can do this. Okay, share screen, go to this. Okay, now you guys can see this. So you can see that you can go to his own paper, you can scroll down to his methods section, and you will find a GitHub link. It's right here. We will click on it. you will again be jump scared by Jeffrey Tompkins. He pops up there in all his glory. You're going to want to go to the chimp contigs, I think. Yeah, chimp contigs. So this is his 2018 18K chimp contigs. Go to chimp contigs on Homo, and you're going to want to download it. Now, when you download it, and I don't know if it's going to actually let me do this. Let's see. Let me see if I can pull it up. Contigs. Jackpot, you guys. Hold on. Pulling it up. And we'll just delete all this data so I can just show you again. We'll just do it live, We'll just do it live. Delete. Okay. Go back over to you guys. I'm going to have to share my screen in like big boy mode here. Present. Stop share. And then I'm going to share screen. It's going to be my entire screen. Like that. Okay. Okay, let me see here. Can you guys see my Excel spreadsheet? If I go like that, can you see it? Okay. I'm going to switch back over to the live and see. You know what? You're just going to have to trust me. Hopefully you've seen it. Okay. So hopefully you guys can see my Excel spreadsheet. And I'm going to check back in a second and make sure that everybody's like, yeah, we can totally see it. So you can see a couple of things here. These are Jeffrey Tompkins' downloaded contigs from his 2018 paper. This is his data, right? All of his data. So the first thing we're going to do is we're going to select all of it, and we are going to turn it into a table. Where's my table? How do I insert? There we go. Yeah, make all that a table. Great. So once everything's a table, you're going to go over and you're going to sort. You are going to sort by length. Smallest to largest. So it's already sorted by length. That's probably because I did this last time and I sorted it by length. Now I'm going to check to make sure you guys are saying that you can see it. Excellent. Excellent. Everybody can see it. That's pretty freaking sweet. Okay. So remember, Jeffrey Tompkins got 84% as his percent similarity for these 18,000 contiguous sequences. This is his published CSV. So we can go equal average and take the average of p-ident. This stands for percent identity. Enter. There it is. There's his 84%. You can see it for yourself. Now, this is unweighted, right? Because what you can see here is that we're just taking an average of the percent identity returns here. So this means that this 200, or let's say this 309, this 444 length here is 69% similar between the chimp and the human. That's like approximately 70% similar. Not very much. It's a really short read, not very similar, a lot of small differences, whatever. But this is being averaged with this sequence, this 167,095 base pair section that is 98% similar. They're being weighted the same. You cannot do this. This would be like being in school and telling your teacher that they need to reassess your grade because you got a 5 out of 5 on attendance on June 6th, right? But then you got a 50% on your test. And you're like, well, I should at least have a 75 because 50 plus 100, if I average those together, then I'm going to get a 75. Right? That should be your grade. No, because the quiz is going to be weighted as like significantly higher than your attendance grade. And so you might be thinking to yourself, well, what if you did weight it properly, right? What if you weighted it correctly here instead of incorrectly? You weighted it by length. When you do that, you actually end up with a 96% similarity. So we take equals, let's see, average, I believe it's an ident here. Oops. Hold on. Equals average in ident equals average length equals, oops. Hold on. Equals first divided by the second. Oops. I think that's the second divided by the first. Equals second divided by the first. Did it a second time. Sorry, you guys. I've been streaming for a hot minute. Okay. It's 96%. When you weight Jeffrey Tompkins' own data by the length of the query and its similarity, you end up with a 96% similarity. That is conventional numbers by his own methods. You can take his exact methods. All you have to do is weight them, and you get 96% similarity. If that's not the biggest like dunk on his own body of work, that you can use the methods of Jeffrey Tompkins, and if you just correct the most wrong thing, you're actually going to get like really close to conventional numbers. So like okay, everybody's telling me how to use Excel. Hush up, you guys. Look, I did my best. I did what I could. I did what I could. The point is that is his data, not mine. That's his data. So like, and the reason I'm emphasizing this is because I already watched this video and I know where this is going. And it's really like super irritating. So I guess let's get back to it. Now that I've taken up enough of your time here with Excel.
The average alignment, identity, or similarity that I achieved from those was 84%. So basically the chimp genome at a maximum is only 84% similar to human, and probably less than that. So what's really interesting is that Richard Buggs, professor of evolutionary genomics at the University of London, Queen Mary, actually did a study himself where he downloaded a DNA sequence that was lined up.
This doesn't matter, and we're skipping it because Richard Buggs published in 2020 a new method of doing comparisons, and he published in a journal, right, that the similarity with his student, he published that the similarity is 96% between humans and chimps, using this sort of it's like an it's like a pictorial means of mapping chromosomes. And what's cool about that, like later in this video, Tompkins is going to say something really, really fascinating with regard to Richard Buggs. And like what I'll tell you kind of ahead of time is that I don't think it's a coincidence that Richard Buggs published the 84 number on a blog and then he published on the 96 number. I don't think that's a coincidence.
84%. Some people would say, well, that sounds pretty similar, even if it's not 98, 99%. Well, the problem is that theoretical evolutionists who are trying to explain human evolution from a chimp ancestor, or an ancestor we shared with chimp, need a 98 to 99% genome similarity to explain human evolution with the known mutation rates that they've actually determined in humans and chimps in 3 to 6 million years, since we supposedly shared a common ancestor with chimps. So for theoretical human evolution from a common ancestor with a chimp to even work, you need something close to a 99% similarity. So 84%.
Okay, please, I'm begging you. Someone show me where anybody has said this ever. Who has ever said this? Who is out here saying that you need to have a 98 to 99% genome similarity to explain human evolution with known mutation rates in 3 to 6 million years? Like, who is actually making this case? I've never once heard this, and I'm getting my PhD in biological anthropology. So I don't know what to tell you. Fellow theoretical evolutionists, please let me know where you've heard this before, because this is new to me, especially given like the conventional number is 96. Like, if this is the case, then the evil evolutionists have already done it. They've already debunked themselves, haven't they? Okay.
Obviously, totally kicks that out the door. So this whole issue of the human genome supposedly evolving from a common ancestor with chimp really begs the question, are genomes really evolving and getting better? Are they gaining information and getting better, or are they actually devolving and losing information?
You guys, we are
what about this whole idea populated?
We are going to skip the entirety of genetic entropy, and I'm going to tell you why. Genetic entropy is nonsense for one simple reason, and it's that they had to redefine the term fitness to even make it make sense. You can't just redefine the definition of like one of the oldest concepts in evolutionary biology and then be like, see, we debunked evolution with this. It's like, well, yeah, I mean, you can debunk anything if you're allowed to just change the definitions to whatever you want. That's like a super ridiculous concept. And then there's also plenty of videos out there of people who have spent a lot of time. Zach Hancock, who was here earlier, did a fantastic video on genetic entropy that just absolutely bodies it into the ground using a lot of the same sources that these guys are supporting for themselves. Ugly German Truce, I'm just looking in the side I see. Where does Jeff get 3 to 6 million years? Look, I don't know. I have no idea where he's getting this, because I've also heard 7 to 9 exclusively. Like, I don't know where he's getting 3 from. And knowing Jeff, I bet you that the work that he's done with these mutation rates shows like it's difficult to get the number of differences in a 96 similarity scenario, that it's difficult to get those differences in 3 million years, which is something no one has said, right? Like no one is saying that. Okay, so we're gonna move on. We're skipping genetic entropy. Other people have done it better than me and to death, and also they had to redefine fitness to make it work. Onward to the fossil
with
record.
Noah's 3 sons and their wives. So what about this whole idea of human evolution anyways? I just want to go over a few slides as to why that's really bogus from an anthropology perspective.
Oh my goodness, you guys, from an what? That's me. I'm an anthropologist, kind of. I'm a student in anthropology. That counts, kind of, right? I've done my own work. I get to be kind of an anthropologist. But now, wait a second. Didn't Apologetics 101 say at the beginning that like people like who aren't geneticists are dunking on Jeff's work and they shouldn't be doing that because they're not geneticists? Well, that's so weird because Jeff's not a paleontologist. He's not an anthropologist, and he's sitting here talking about human evolution. Yeah, that's so strange. Boy, that almost feels a little hypocritical, doesn't it?
So this is typically a typical figure that you might see, and basically you see apes there at the start, you see humans there at the end, but essentially everything in between is hypothetical and theoretical. That's all we really see in the fossil record anyway.
Wrong, Jeff. You are wrong there. We have Sahelanthropus chilensis, Aurantuganensis, Ardipithecus cadaba, Ardipithecus ramidus, Australopithecus anamensis, Australopithecus afarensis, Australopithecus barroghazali, Australopithecus diarmida, Australopithecus garhi, Australopithecus africanus, Australopithecus sediba, Homo habilis, Kenyanthropus platyops, Homo rudolfensis, Homo erectus, potentially Homo georgicus, Homo erectus, potentially Homo ergaster, Homo erectus sensulato. You have Homo naledi, Homo floresiensis, Homo heidelbergensis, Homo rhodesiensis, Homo bodoensis, Neanderthalensis, Homo neanderthalensis, Denisovans, and Homo sapiens. There are quite a few guys in that list that I just said that span this sort of in-between section here that I don't think our pal Jeff here is gonna touch on. But I do know he's gonna touch on one of them. It's Lucy. Surprise.
Is humans and apes. So let's start with Lucy. So commonly known as Australopithecus afarensis. The skeleton was only about 40% complete. That's all they had of it, and it was discovered in 1974.
Okay, 40% complete, like of a full skeleton. Lucy is bilaterally symmetric. So like there's going to be a lot of that 40% that overlaps down the midline. Now, if they were completely, like hypothetically, two completely different, like a unique 40% that doesn't reflect, you might get, I don't know, a max of like 80% of the skeleton. But it also depends on how you're comparing, like how you're counting up the skeleton. Are you doing it by bones? Like, I don't think you need every rib to get a decent idea of what the animal looks like. Or are you doing it by mass? In which case, I think it's going to be significantly larger than 80% even, because if you look at how much of the skeleton is there, a lot of the bulk of the skeleton is going to come from like the pelvis, the vertebral column, the skull. Primarily the skull is missing in Lucy's specimen. You've got that right humerus. You've got a lot of the left humerus. You've got a lot of the right and left forearms. Also, you're missing a lot of the hands and feet. You're missing the left femur. You're missing most of the fibula, kind of the shaft of the tibia. But you got quite a lot going on here. Let's let him tell us why it's bad, though.
They dated Lucy at about 3 million years old and claimed that she was bipedal and walked upright. However, we've got major problems with this, admitted by the evolutionists themselves. Did Lucy really walk upright? So Eric Stockstad, an evolutionist, published this article in Science in 2000, and he said, I walked over to the cabinet, pulled out Lucy, and Shazam. She had the morphology that was classic for knuckle walkers. So in other words,
okay, well, you know what? Let's let him finish here.
When Lucy wasn't swinging around in trees, she was on the ground, walking around on all fours like chimpanzees do, because they're designed to be knuckle walkers. They have special locking wrists that humans don't have, so they can walk around and do that. And there's a picture of a chimp doing what chimps do, knuckle walking when they're on the ground.
Okay, so Stockstad and also Brian Richmond are two of our big guys who were these knuckle walking proponents for Australopithecus afarensis back in the early 2000s, so 2000, 2001, etc. I believe Straight was also involved on this. And they do it based off of some particular notching that's going on in like the carpals and the distal end of the radius. This has not stood the test of time because a lot of the knuckle walkers that they assessed in their study, and this was noted by, I think it was Lovejoy and colleagues later on, a lot of the specimens that are known for knuckle walking are actually like missing the notch, right? And a lot of the organisms that some of the organisms, I believe, some of the organisms that are not knuckle walkers had partial notches. So it doesn't do a good job. Like the primary characteristic that Brian Richmond and that Stockstad and that Straight were relying on to distinguish knuckle walkers from non-knuckle walkers is not actually a diagnostic characteristic in the carpals. And I also know this because one of my colleagues actually works on hands. She studies hominoid hands, so the hands of extant gibbons, ape, like all of the great apes, so like Pongo, gorilla, panins, and humans. And she actually published, like she's published on the material of like hominoleti before. Like she does this. She pretty much knows hands like very well. Chimpanzees and gorillas knuckle walk in different ways. So that absolutely wrecks this idea of a knuckle walking ancestor for the sake of parsimony. They've actually evolved knuckle walking uniquely because the mechanism is actually separate. And that would also explain why the singular notch isn't very good at distinguishing and discriminating knuckle walkers from non-knuckle walkers. But more importantly than all of this is that Lucy's entire morphology is that of a biped and could not be that of a quadruped. Period. Biomechanically, it does not work. The foramen magnum is anterior and the position is forward. That's what you need for the foramen magnum to actually to characterize a biped to the exclusion of other types of locomotion within primates. She has a bowl-shaped pelvis with sagittally oriented iliac blades, and it has an anterior inferior iliac spine, which is a critical muscle attachment site for maintaining stability, being upright. And it strengthens the pelvic floor, the bowl shape of the pelvis, not the spine, not the anterior inferior iliac spine. I mean, she had a valgus knee, the carrying angle that allows you to hold your weight in a singular column. She had the inline big toe and the 3 arches in the foot. And what Tompkins is going to go on here, so he's going to be like, oh, we didn't find any hands and feet with Lucy, and the Laetoli footprints are far away from Lucy. So arguing that Lucy's kind made these footprints is ridiculous. No, it is not, because we have Australopiths that are dated to the time period of the Laetoli footprints, and Australopiths are the only thing living at that time that has the foot morphology that matches that we see at the Laetoli footprints, a condition that is intermediate to how chimpanzees walk bipedally and how anatomically modern hunter-gatherers, unshod, walk bipedally. Now, then he complains about the nature of the recreation for Lucy. We could corroborate that with other Australopiths that they will never bring up. They're not going to talk about Australopithecus sediba and how we have the entire half of a pelvis for Australopithecus sediba that corroborates the reconstruction of Lucy. They're not going to talk about STW 573, which we have almost 100% of that skeleton, including the foot that shows it has an inline big toe and suggests the 3 arches in the foot as well. They're not going to talk about the Dakika child. They're going to skip all of this, because they can't deal with it. They can't actually fit it into their model because it patently denies quadrupedalism for these hominids. It doesn't work. Okay, sorry, spiel over.
And so this is another quote from an evolutionist. And I love the name of this article, Lucy on the Ground with Knuckles. And this was published in Science News in 2000. And they said Australopithecus anamempsis and skeleton known as Lucy had wrists capable of locking the hands in place during knuckle walking. Walking Ancestor Nature 2000. And these evolutionists said, regardless of the status of Lucy's knee joint, which they can never come to a conclusion on, new evidence has come forth that Lucy had the morphology of a knuckle walker.
That is absolutely patently false. They did come to a conclusion on the Australopithecus afarensis knee joint. It's valgus. Like he just added that in. He's like reading a quote and then he adds his own little tangent in there to make it sound like it's a part of the quote. Now, Richmond and Straight, to my understanding, even in this, and you'll notice I mentioned them earlier, right? Like I've seen this video, but I didn't need to see that to know who is known for this knuckle walking hypothesis. And they have not published, to my understanding, on, like Richmond hasn't at least. He's not holding to the idea of a like walking mechanism for Lucy anymore. And like I did a brief literature search this morning and I couldn't find anything in his more recent works, like his 2016, he like had published a chapter on hominid hands. And he doesn't talk about Australopithecus afarensis having a walking knuckle, but he does talk about knuckle walking and he does talk about Australopiths. So it's kind of weird that he's not kind of seemingly holding to that opinion anymore. But even then, Richard and Street didn't think Lucy was knuckle walking 24/7. They think that it's basically vestigial. It's a mechanism that's still it's still around. And part of the reason for this is that Lucy doesn't have the rest of the morphology that's going to be conducive towards knuckle walking, as we just discussed. But also, her hands look more like human hands than they do like chimpanzee hands. So she's got a long thumb and short fingers instead of the opposite.
And this is a great quote from 2016, and the title of this paper says it all, really. From Australopithecus to Homo, the transition that wasn't.
Oh my God. Oh my God. Jeff, this paper is about how the transition is too smooth. It's talking about how the transition didn't occur because it was so slow. Jeff, he didn't read the paper. Of course he didn't read the paper. Why would he read the paper? Although the transition from Australopithecus to Homo is usually thought of as a momentous transformation, the fossil record bearing on the origin and earliest evolution of Homo is virtually undocumented. Hey, you guys, let's read that paper. Let's look at that quote real quick, because I'm pretty sure Donnie has made this same mistake. Australopithecus to Homo, the transition that wasn't. This is like a pretty classic paper. Let's see. We need to get our PDF on here. Please be. Yes. Okay, beautiful. Control F, momentous. Okay. Let's check it out. Let's check this out. We're just gonna like read do a little abstract readerino here for our own purposes, for our own nefarious evolutionary purposes. And where are you? There you are. Okay, so here's the paper. From Australopithecus to Homo, the transition that wasn't. Although the transition from Australopithecus to Homo it's the first line of the abstract. Oh my God. Oh my God. It's the first line of the abstract. He couldn't even read past that. Although the transition from Australopithecus to Homo is usually thought of as a momentous transformation, the fossil record bearing on the origin and earliest evidence of Homo is virtually undocumented. As a result, the poles of the transition are frequently attached to the taxa Australopithecus afarensis and Homo habilis or Homo erectus, in which substantial adaptive differences have accumulated over significant spans of independent evolution. Such comparisons, in which temporarily remote and adaptively divergent species are used to identify a transition, lend credence to the idea that genera should be conceived at once as monophyletic clades and adaptively unified grades. However, when the problem is recast in terms of lineages rather than taxa per se, the adaptive criterion becomes the problem of subjectively privileging key characteristics from what is typically a stepwise pattern of acquisition of novel characteristics beginning in the basal representatives of a clade. This is the pattern inferred for the species usually included in early Homo, including Homo erectus, which has often been cast in the role of the earliest human-like hominin. A fresh look at brain size, hand morphology, and earliest technology suggests that a number of key Homo attributes may already be present in the generalized species of Australopithecus, and the adaptive distinctions of Homo are simply amplifications or extensions of ancient hominin trends. Dude, you can't get away with this, John. Why did I say John? I had like a Sanford moment. You can't get away with this. What is with these guys and not reading past the first line of the abstract? Okay, now this is the one that I was thinking of from our pal Donnie. Conclusion. The fossil record bearing on the ancestry of Pleistocene Homo is poor. However, the more we learn about early Homo, the less compelling is the case that an adaptive shift can be read from currently documented skull and skeletal anatomy as a major transition from generalized Australopithecus precursors. Like the transition that wasn't, because it was so slow and gradual. It wasn't like this big, massive, momentous event. So this is what you call a classic Jeff move. Not a good look, I would say. So let's hop back to him since this is all about Jeff anyways. Share screen, back to, there it is.
So they said in the paper, although the transition from Australopithecus to Homo is usually thought of as a momentous transformation, the fossil record bearing on the origin and earliest evolution of Homo is virtually undocumented. And that was in the Royal Society 2016, and nothing has changed since then.
Jeff, you didn't read the paper. You didn't read the paper. You don't get to quote the paper when you didn't read it.
So what about this whole idea of Neanderthals and so-called primitive archaic traits in humans? The fact of the matter is you wouldn't even know a Neanderthal unless he came up to you with a name tag and a spear on the street because they looked human. And in fact, now they're sequencing DNA, human DNA. They're sequencing DNA from Neanderthals that have been actually ritually buried in caves because they're human. And so we see stories like this in the news quite frequently now. Humans.
That's going to be a really tough sell for Jeff when these big announcements that are supposed to come out tomorrow by Lee Berger and company that I have a very strong feeling are going to involve the ritualistic burial of the dead at the Homonoleti site, a hominin with a brain case the size of an Australopithecus. Like, burying your dead makes you human. AIG is going to have to do a 180 on Naledi real quickly.
Bred with this mysterious species more than once, new study shows. Your Neanderthal DNA might actually be doing you some good. So yes, there are evolutionists out there saying we need to clone Neanderthal brain genes and put them in our own genome because Neanderthals had bigger brains.
Name one.
So that doesn't sound like evolutionary thinking to me.
We didn't descend from Neanderthals, Jeff. They're a cousin. They're a cousin.
Ancient humans may have been mothers to some Neanderthals earlier than we thought. So this is the general consensus that humans and Neanderthals interbred, and essentially Neanderthals, according to evolutionists, were archaic humans.
Wrong. Absolutely 100% wrong. They're considered a distinct species on the basis of morphology and on the basis of their genome, which falls outside the human range. Not only that, but I think it was Tattersall's paper. Like, if we were being consistent with speciation, like we were using modern morphologic standards and applying them to the past, paleoanthropologists like Ian Tattersall, I think it was Tattersall, have proposed that like Neanderthals would hash out as a different genus. That is how distinct some of their craniofacial characteristics are. Now, I don't think that should be done. I really don't. I think that they were incredibly similar to humans, but they're not the same species. And I don't really know anybody who holds to the idea that they are.
But the fact of the matter is they were just humans. So the Neanderthal skull was typically larger than a modern human. They had a sloping forehead and a enhanced brow ridge, or pronounced brow ridge. And in fact, the Homo erectus skulls, which is another class of archaic human, their skulls were not as large as Neanderthal skulls. They were a bit smaller, but they had essentially the same feature, the sloping forehead, the large brow ridge. But
okay, Neanderthals have uniquely derived characteristics. They have the occipital bun, the retromolar gap. They have the platycephalic skull, right? These guys are not, they have different wear patterns on their incisors. That's not, I mean, it's morphologic, but it's also behavioral. They are not, they're not going to be like clockable as Homo sapiens in the fossil record. Like, they are distinct. They've evolved their own distinct characteristics. And Homo erectus isn't going to be confused for Homo sapiens or Neanderthals because of its characteristics that are diagnosed.
This is really an archaic trait. So there's a dentist in South Africa who takes pictures, who's a Christian and a creationist, takes pictures of his patients that have these so-called archaic traits. And here's a guy that was in, I don't know if he's having a cavity filled or what, but anyways, he got his picture taken. Look at the brow ridge on this guy. And
does he have an occipital bun and a retromolar gap?
He is a young man and alive and well. I took these pictures of rugby players in Eastern Europe, which it's interesting because Neanderthals are typically found in Eastern Europe, also in other parts of the Middle East and some other places. But anyways, look at these guys. They have sloping foreheads and big brow ridges. This is a famous guy, Nikolai Valuev, a former professional boxer. He's a member of the Russian parliament now.
This picture exists in like the published like PDFs that Ramat and Standing for Truth put out. Like, all of these guys just constantly crib from each other. They can't come up with anything original. Yeah, this guy has a really big brow ridge. Guess what? A brow ridge isn't a diagnostic characteristic of a Neanderthal, right? Like, you need to utilize the characteristics that are only found in Neanderthals if you want to say that they are found just as within the normal human Homo sapiens variation, which you're not going to be able to do.
He has a sloping forehead and a huge brow ridge as well. So this really isn't an ancient archaic trait. It's actually found among modern humans today in different parts of the world. But what about this whole idea of a sagittal crest, or this ridge along the top of some people's heads?
No,
that evolutionists want.
A sagittal crest isn't diagnostic of any of the hominins like post habilis, right? Like, what is he saying? Okay. if he really wants to make this case, what he needs to do is he needs to show that diagnostic characteristics that are exclusive to Homo sapiens can be found in Neanderthals, like the vaulted forehead, the globular shape to the skull, the chin, things of that nature, that those can be found in Neanderthals, and that the Neanderthal diagnostic characteristics can be found in modern humans, any of them. Retromolar gap, occipital bun, the lack of the chin, the platycephalic skull. Notice the sagittal crest isn't in any of these groups as like a diagnostic characteristic. And also, this isn't a sagittal crest. Like, at best, you've got like a keel going on here.
Once claimed it was archaic, and so the arrow is pointing to a guy there with a pronounced sagittal crest, but the guy up on in the stute there is a famous neurosurgeon, and he has a sagittal crest, and he's a pretty smart guy. And then, of course, the other guy over there, he pilots a star cruiser across the galaxy, so he must be pretty smart. But the fact of the matter is, these so-called archaic traits are really found among modern humans. There's just a lot of variation among humans. So here's two.
Okay, there is a lot of variation in humans, and humans don't have a lot of genetic diversity. So why then do Neanderthals fall outside of our genomic variation? They fall outside of our variation of our range genomically and morphologically. Both of them. Like, and these guys are going to have even more trouble once they talk about Homo erectus, right? I don't know if Tomkins is going to get into Homo erectus very much here, but he alluded earlier to the idea that Homo erectus is like this hump, this archaic human. That's the wording that he used. Okay, what are you going to do about the diminutive statured 600 cc brain case size, like very prognathic faced Homo erectus found in Dmanisi, Georgia? Are those human? What about Homo habilis? Is that human? Right? Where do we draw the line here? And the answer is the line can't be drawn because there's no characteristics that actively distinguish it really any of these hominins in such a way that can be unless they're like we're looking at like apomorphies here. None of these characteristics are going to be able to lump these groups into like the human group as a diagnostic characteristic and the ape group as a diagnostic characteristic. That's what I was trying to say.
Two professional tennis players. This was actually a very close match, but the one guy is almost 7 feet tall and the other guy is 5'7". So anyways, there's just a lot of variation among humans for different traits, and it's variation, not evolution.
What? You see what I mean about they don't know what fitness is? Like, this makes sense that Tomkins was talking about genetic entropy earlier, right? Because like variation exists and can be selected upon to increase fitness in a given environment. Like if that allele moves to fixity or to fixation, excuse me, like that is evolution. So no, human variation is not like actively evolution, but the variation can become evolution if it's selected upon. And that variation is the result of mutation, which is a mechanism of evolution. So I guess it kind of is evolution as well.
All the other animals that God created, humans were created in God's image. So the Bible says, so God created man in his own image. In the image of God, he created him, male and female, he created them. So humans were the pinnacle of creation and created in God's image. And this is why, in my opinion, human evolution is one of the top evolutionary dogmas, because they're really attacking Genesis and the nature of man fundamentally at the beginning of the scriptures. So why is this important? Well, it has to do with the idea of a historical Adam. Did Adam really exist, or did we evolve from apes? And of course, there's an organization that is theistic evolution at its core. It's Biologos. And you might as well be on the Berkeley University website reading about evolution, because you'll get the same story. This is a problem.
That was such a pot shot, wasn't it?
Because the Bible has very distinct and clear genealogies and chronologies back to the original Adam. Why is that important? It was brought into the world through Adam and Eve when they fell at the beginning in Genesis, and that's documented in Genesis chapter 3. But the Apostle Paul clarified the importance of this in his epistles. In Romans 5:12, he says, therefore, just as through one man sin entered the world, and death through sin, and thus death spread to all men because all sin. So in other words, through one man, meaning Adam, sin entered the world.
Jeff, please, we know the
sin,
spiel.
so even, and I talk about the human chimp DNA similarity in this gene.
Showing for his book, we are going to skip the book
and
so on Amazon.
All right, awesome. That was great presentation. I do plan on doing some videos later on Australopithecus and things like Neanderthals and things like that. So I appreciate that.
Don't do it.
But today we're primarily going to be focused on your particular methodology. And I have some questions because I don't think I heard anything on chromosome 2 fusion in your presentation. Could you explain that real quick before we jump into the other stuff?
Oh my God.
Yeah, I actually have done entire presentations on that can get very technical. So in the so-called chromosome 2 fusion, evolutionists believe that 2 chromosomes fused from the ape genome to form human chromosome number 2. The reason they had to go that route was because humans have a chromosome number, a diploid number, 46 chromosomes, and apes have 48 chromosomes. And so they did find some gross similarities doing cytogenetic spreads.
Okay. Oh God, do we really want to do the human chromosome 2 stuff? Do you guys want to do this? Tell me in the chat. Are we doing human chromosome 2? You guys let me know. I don't know if we should, but we can. We can gear in for some human chromosome 2. Should we? Should we do chromosome 2? Okay, I'm going to make a poll here. Hold on. I know I can do this. Start a poll. Do we do chromosome 2? Okay, I asked the question. I'm going to go pee, and you guys let me know when I get back. Okay. Back. Did you guys like being left alone with the skulls? I know you did. I know being left alone with the skulls made you all very happy. Okay, time to check out this poll. Let's see what the peanut gallery said here. I feel like I already know what you guys said. Yes, 74%. Okay, we're doing it. We will do it. Okay, you guys, we're doing chromosome 2. We are going to discuss chromosome 2. How do I want to do this? I think what we're going to do is we're going to pull up human chromosome 2 fusion. I think Ken Miller has a really awesome diagram for this. So we're just going to do that, and I'm going to show it to you, and we're going to talk about it. Okay, present, share screen, initiating screen share, going over to human chromosome 2 fusion. Okay, all right, so human chromosome 2. You guys, back in the day when we first did karyotypes for the great apes, it was something of a surprise, I think, that humans had 46 chromosomes and the great apes had 48. The other great apes had 48. So a prediction was made in like 1972 by Turlo and Grouchy, where they were like, okay, well, like one of two things happened, right? Either all of the other great apes, like the ancestral condition was human-like and it had 46 chromosomes. Some Miocene ape had 46. And then in each great ape group, they subsequently experienced some kind of fission event to give them 48. Or alternatively, the opposite, more parsimonious answer, which is that 48 chromosomes is the ancestral condition and modern humans experience some kind of fusion event. So with this prediction set, they were like, a fusion event probably occurred, and based on like kind of brute banding, they were like, it was probably in human chromosome 2 for two reasons. One, because human chromosome 2 is big, and two, because human chromosome 2 has remarkable synteny or similarity with chimpanzee chromosome 2A and 2B, which at the time were known as 12 and 13. So what you're looking at here is sort of the hypothetical situation here. We have our ancestral chromosome here and our other ancestral chromosome here. And if a fusion occurred, there's going to be some like pretty classic calling cards to let us know, like a smoking gun, if we're looking at a fusion site. The first thing is we should find a fusion site. It should be in the middle of the chromosome. So like, how do you find a fusion site in a chromosome? The answer is actually really easy because the ends of chromosomes are capped with what are called telomeres. Telomeres are highly repetitive sequences of DNA, and they protect the chromosome during things like division from fusion, basically. And they also protect the genetic material that's kind of further upstream from these telomeres. So you should find a fusion site by finding telomeres in the middle of a chromosome at the fusion site. And these telomeres are clockable on the basis of a highly repetitive sort of telomeric motif that is GGG, TAA and forward, and CCC. No, it's hold on. What is it? What is it? Telomeric repeat. It is, someone just tell me. It's GGG, TAA, and CCC, ATT. Whatever. It's a telomeric repeat. I just did a video on this and I can't freaking remember it. So there's telomeric repeats that as a motif can be found in forward and in reverse in high densities in the middle of the chromosome if a fusion happened. That's the prediction. And secondarily speaking, there should be a second centromere, right? Because a chromosome has one centromere. So if chromosome 2 is the fusion of two ancestral chromosomes, there should be two centromeres. So there should be a telomeric calling card in the middle of the chromosome and two centromere calling cards. These are alphoid repeats. That's what they're known as. And so we have found this in like staggering detail, right? We have a 798 base pair fusion site located in the middle of human chromosome 2 that is degraded telomeric repeats. The fact that they're degraded is super important because chromosomes can't experience fusion with pristine telomeres. They would prevent fusion. So the fact that there's degradation going on means the telomeres in this ancestral condition were absent or failed in some way and resulted in a fusion site and then rapid degradation on top of that. So you've already got subtelomeric regions, which are kind of like, worse versions of telomeres with regard to the sequences. So we found the fusion site, 798 base pair long series of telomeric repeats. We found the second cryptic centromere, and it maps to chimpanzee 2B. And when I say it maps to it, I mean it shares the genes upstream and downstream of this proposed cryptic centromere that map to the genes around the normal chimpanzee 2B centromere. Those two things map. Right, so we've got both of those, and then moreover, the most important thing, in my opinion, is the overall synteny. The chimpanzee 2A and 2B share 98% approximately percent similarity with human chromosome 2, and I know this because I tested it, and you're going to get this in a human chromosome 2 video that's coming up. But what I did is I did a comparison of chimpanzee 2A and 2B to human chromosome 2, and I did full coverage. Every single base pair is covered. So it's 98%. It's actually like 98.2, 98.8, depending on which section you're looking at. So they're remarkably similar. Now, the arguments that creationists use against this, primarily what Jeff does is he says the argument against the fusion site is that there is a functional gene spanning the fusion site. He says across the middle of the fusion site, there's this gene called DDX11L2, and DDX11L2, it performs all these critical functions. You couldn't possibly live without it. Anyways, Glenn Williamson, who Tomkins hates, actually also did some work on this that I found and utilized in my chromosome 2 video that's coming out soon. And it turns out that DDX11L2 is expressed about 5% as frequently as normal genes. So not exactly critical. And then also it doesn't span the fusion site in every transcript. It spans the fusion site in approximately 21% of transcripts. And I know all of that off the top of my head, but I couldn't tell you the telomeric repeat, which makes me look like a big dummy. So those are the two primary things that's the primary argument that Jeff is going to use against the fusion site. And the fusion site is in fact very remarkable. So someone is going to tell me, hopefully, please, someone in the chat, did no one correct me? You guys are so sweet. You guys are so nice. You can correct me, though. I do deserve it sometimes. I need to get busted when I make my own mistakes. Okay, back to the video. Let's hear Jeff out.
Of human and ape chromosomes. But they also found some problems with it as well. But anyway, so that's what led to the whole idea of it. And then they thought they found in the early 1990s actually a DNA sequence that represented the fusion site, which they claimed was a head-to-head fusion of human telomeres. And so it was a very small signature. It was only about 800 bases long. And the reason I say small is because human chromosomes are 5,000 to 15, or I'm sorry, their telomeres at the ends of them are 5,000 to 15,000 bases long. So if you have two telomeres allegedly fused together, you should have a signature of 10,000 to 30,000 bases, not
No, you shouldn't, because remember, degradation of the telomeres is like a prerequisite for fusion to occur. You wouldn't get pristine head-to-head telomeres in like the same identical densities to those at telomeres, because if those densities existed, the fusion wouldn't happen. Jeff is a geneticist.
800. And so on top of that, the fusion site is very muddled, if you will. It's evolutionists would call it degenerate. In other words, it's not a pristine head-to-head fusion. It's only about 70%, similar to a pristine fusion of the same size, about 800 bases. So anyway, so it isn't that clear cut. And on top of that, the fusion site is actually found inside a long non-coding RNA gene. And if things couldn't get any worse for the evolutionary fusion story, the fusion site is actually a second promoter or a switch inside this gene. It binds RNA polymerase II, which initiates transcription in the middle of the fusion, so-called fusion site. All sorts of transcription factors bind to the fusion site. And I basically mined all these ENCODE data sets to verify all of this.
So
and if you look
again,
on a genome browser, you will see the fusion site inside at the beginning of this gene. So
okay, so again, when he's talking about the critical nature, how often it's transcribed, how important it is in all these tissues, like again, it spans the fusion site in roughly 21% of transcripts, and it's expressed overall about as much as or about 5% as often as your average run-of-the-mill gene. So no, none of this has any bearing on the fusion site being a fusion site. But moreover, genes can also move around in the genome. So even if it was an absolutely critical gene that had an imperative function, it is possible that that gene existed in the ancestral condition as well and just moved over the now somewhat free real estate fusion site. Like that is also possible. But you don't even need that explanation because it's not a functional gene. It's actually categorized as a pseudogene. Now let's see if he actually tells us the real punchline of this gene, which if you know the real punchline, say it in the chat now.
Any genes in the human genome have multiple promoters or these switches. A lot of these switches are in front of the gene, and a lot of the switches are inside the gene right at the beginning, maybe after the first exon, like the fusion site is. So anyways, it is actually a functional feature inside a gene. And this gene is an important gene. I actually looked at the expression of this gene. It's expressed in many different tissues in the human body. It's connected with the expression of other genes in top-level networks, including the formation of blood cells. And I actually annotated the messenger RNAs that were produced from this particular gene that the fusion site is in. And these messenger RNAs are full of all sorts of RNA binding sites and all sorts of functional features. So it's not an evolution might say, oh, it's just spurious transcription, you know? No, it's a real gene that's producing real transcripts that do real stuff, important stuff. And
it's a real pseudogene that is transcribed spanning the fusion site 21% of the time and is expressed about as often as 5%, or is expressed 5% as often as other genes. He's still not saying the punchline though. He's not telling you the most important part about DDX11L2. Let's see if he says it.
And so the fusion site is bogus. It is a feature, a functional feature inside a gene. And so I can get really deep in the technical muck with this. And I actually have an entire talk that I gave at the International Conference on Creation in 2018, where I actually developed some software to scan the human genome for internal telomere sequences and then line up those genome coordinates with all these all sorts of internal telomere-like sequences that are associated with a variety of functional aspects and all sorts of things. So anyways, so that, and actually published a paper on that. The whole fusion scenario is not viable.
Tell them about DDX11L2, Jeff. Tell them about the gene family it belongs to.
When you begin looking at the genetic data, in fact, I even looked at the so-called cryptic centromere site, which is less convincing than the fusion site. So if you had two chromosomes fused, because each chromosome has its own centromere, theoretically you would get two centromeres.
He's going to skip it. Okay, I'm going to have to tell you guys the punchline since I guess Jeff isn't going to do it. And I watched this. I don't remember if he actually says it. So I'm going to spoil it for you. But to do so, we're going to need to hop back over to our little we're going to need to hop back over to our images over here. Okay, you guys. So DDX11L2 here's where it is, right? It spans the fusion site. Let's just take the best case scenario for Jeff, right? It spans the fusion site. It's here in the middle, located in the middle of this fusion site, this 798 base pair long sequence of DNA in the middle of human chromosome 2. It's a pseudogene that belongs to a family of genes in the DDX11L family. You want to know where every other member of this gene family is found exclusively? In telomeres. It's in telomeres, you guys. They're all found in the telomeres of chromosomes. Here, 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 here. So what's a telomere-specific pseudogene doing in the middle of human chromosome 2? I ask you, you people, what is it doing there? Can you tell me? It's because it's a fusion site. It's a fusion site. And Tomkins got asked this question in a different video. Let me see if I can actually find it. It actually might just be worth looking at here whilst we're already with one another. So let's see. It was in Logos Associates, I think. Tompkins. We could. Oh please. Okay, here it is. Here it is. We'll see if we can get to it. I'm gonna skip around until I find what we're looking for here, because it was in the question and answer section. Then I'm going to share the site. I'm in this, hold on, Telomere, maybe? Down here. Shoot. I'm not going to be able to find it, am I? And then I'm going to look like a fool in front of all of you. That would be so embarrassing for me. Okay, wait. We might have it. Okay, sweet. Found it. Let me switch over. We're enjoying our Jeffrey Tompkins-a-thon here. Jeffrey Tompkins stream-a-thon. This is he's the only one to ever get a person-specific stream-a-thon, so maybe he should be grateful for that. Okay, monkey business in the human genome. Okay, here it is. This is a different Jeffrey Tompkins video, but our friend Dan of Creation Myths asks a very interesting question about the telomere-specific pseudogene found in the middle. Let's see what he says.
This is from Creation Myths again. Why are all the other instances of the DDX11 pseudogene family in subtelomeric regions? And would you mind maybe explaining that for our lay audience?
Yes, I am aware of that they are in subtelomeric regions. Subtelomeres actually have lots of genes and in them, and they're actually very, very important regions of the genome. Lots of gene activity going on.
Yeah, and what about them being found in telomeres? Why is there a telomere-specific gene in the middle of human chromosome 2, Jeff?
On there. No, I don't know why there's so many in those regions. And then you also have the protein coding DDX genes as well, which are not.
He doesn't know. He does not know, you guys. So I don't know what to do with this man. I don't know what to do with them. I'm getting so worked up about this. It's just, what do you do with this kind of information, right? Like Jeff is an actual geneticist, one that is highly lauded and praised by all these young Earth creationists who think non-geneticists don't get to weigh in when Jeff he can't actually have a conversation on the topic that he is debunking. And you can go back and he doesn't answer the question. You can go back and watch the rest of that, but we are here to watch a different video. So he does not answer the question at all. Why? There is a telomere-specific pseudogene in the middle of human chromosome 2. Oh, dang it. Okay. All right. Of course, I talk all this trash, and then I can barely manage to fart my way around StreamYards. Okay.
And then one of them would supposedly be deactivated after the fusion. So I actually looked at the chromosome 2 cryptic centromere site, and it was also inside a gene. In fact, it was in a huge protein coding gene that produced a transmembrane spanning protein that after protein is produced to be transported to the cell membrane and work as some sort of a receptor with the part of the protein sticking outside the cell, the other part sticking inside the cell. And so was actually inside this large protein coding gene. And in fact, part of the so-called cryptic centromere actually coded for the actual resulting protein from the processed messenger RNA after it was transcribed. So
okay, so remember the cryptic centromere, the genes that surround the cryptic centromere in human chromosome 2 are like analogous to the genes found around chimpanzee centromere 2B. What are the odds? You guys, what are the odds?
And on top of that, the so-called cryptic centromere actually had human-specific repetitive elements in it that you don't even find in apes.
There are specific repetitive elements like that are species specific, right? Like he's trying to draw this dichotomy. You find this stuff in humans and you find this stuff in apes. There is no way to do that, right? If you want to say you find human specific stuff, then whatever that thing is that you're looking at, take orphan genes, for example. There are human specific orphan genes. There are also chimp-specific orphan genes and gorilla-specific orphan genes and orangutan-specific orphan genes that are only found in them. But there are no orphan genes that are ape-specific that don't include humans. You see what I mean?
And so the whole cryptic centromere site thing was even as problematic as the so-called fusion site. So any papers on that, maybe you could put some links up to those. I can send you those links later.
Yes,
So in a nutshell, that's
yes, I'm gonna link all your peer review.
In a nutshell, in a nutshell, you guys, that's it. That's why it's not a fusion site. Cool.
Papers that I find on Answers Research Journal. I'm gonna link those in the description box. I have your book right here, and you do make an excellent case against the idea of fusion. You mentioned the 798 base pair length, which is really small compared to where it should be. Also, you got end-to-end fusion. You mentioned that as well in your book with telomere and telomere, which doesn't happen. Is that correct?
Yeah, the only time we actually see telomere fusions is in cancer cells, which are basically gone hog wild and are not healthy.
Jeff, please don't make me do this to you. Please don't make me do this to you in front of everybody right now. Please correct yourself here. Let me give him a chance to correct himself here. He's saying there's no such thing as telomere fusions except in cancer cells. Let's let him finish.
We do not see actual telomere fusions in nature. We see other types of fusions, but they don't involve two telomeres coming together.
Right.
And so even if we look at what happens in nature, that doesn't help the story either. And of course, chromosome fusions in nature are pretty rare. They have happened on occasion, but they are pretty rare. But they do not involve two telomeres fusing together. Sometimes they involve a telomere, but the other side of the equation is not a telomere. It's another type of DNA sequence. And in fact, I actually published a paper on human telomeres and showed how well designed they are. So the DNA at the ends of chromosomes, these telomeres folds back into itself, and it's covered and protected with a protein complex with multiple proteins in it called the shelterin complex, which not only protects the ends of the chromosomes from fusing, but it's also a dynamic machine, if you will, that interacts with the cell cycle, and it assists in cell division in various aspects of that. And so it's actually the ends of chromosomes are actually very, very well protected and very vital to the healthy function of a cell.
Okay, everybody, you heard it from him first. There are no instances of telomere fusions in other animals, and he said this also in his other interview. This is something that Jeff is patently, blatantly incorrect on. But here he is, a geneticist publishing confidently on the topic. So let's take a little stroll here and let me, well, since I was pulling up the the papers in the background, we'll take a little look-see, and you can let the your old pal Gutsick Gibbon walk you through what's going on here. We're gonna start with pigs. So I have here a paper that is titled, hopefully you can see it. Let me just double check to make sure you can see it. Yeah, there it is. This is titled Recent Fusion Events During the Evolution of Pig Chromosomes 3 and 6 Identified by Comparison with the Babirusa Karyotype. So here we are on our screen share. We'll just look at the little section in the abstract here. Likewise, we conclude that pig chromosome 6 was most likely derived by a telomere fusion of ancestral chromosomes homologous to the Babirusa chromosomes 6 and 14. This is a paper from 1996. He should know about this, right? But we don't have to just talk about that one. We can also talk about horses and zebras, the equids, and it is within zebras that we also find telomere to telomere fusion. So let's take a look at that. Oh, and by the way, there is an additional paper from 2012 that has come out since that other pig paper that does in fact confirm that there are telomere fusions there. Okay, so here we go. Insertion of telomeric repeats in the horse and human genomes, evolutionary perspectives, telomere. Let's do fusion. fusion. Okay, Robertsonian translocations, ITSs, subtelomere confusion, ITSs in humans. Let's see where we're going here. Oh, shoot. You know what? Sorry. Hold on. Wrong paper. Let me fetch the correct one for zebras here. I won't let you down. Don't worry. I will not let you down unless I do let you down. That's porcine. Ah, here it is. Got it. Copy, paste into the right location. That's me. I'm doing that to Jeff. I'm bopping him. Okay. So here we are. Robertsonian fusion and centromere repositioning contributed to the formation of satellite-free centromeres during the evolution of zebras. We'll control F for fusion, and we'll go down to the telomere section. Actually, maybe telomere would be easier. Yeah, there it is. EBU8 derived from a telomere fusion between elements orthologous to the acrocentric ECA31 and the metacentric ECA4 at a cytogenic level. It's derived from telomere fusion. No telomere repeats detected. So, you guys, what was it that Jeff said about how there are no telomere fusions located in any other animals? And like, here's the part that sucks about this. Here's the punchline to this portion. And I'm giving the what is it, the goose away with the eggs here. I'm doing something. I'm doing some kind of analogy or metaphor here. Creationists accept that babirussas and pigs are related, and they accept that zebras and horses are related, and they accept that those two groups descend from an ancestral animal, which means categorically they accept some telomere fusions. They have to, unless babirussas and pigs are separate kinds, or horses and zebras are separate kinds. So they accept that fusions occur. So not only do they happen, but they have to accept, if they think that those are the same kinds, that all suids are the same kind and that all equids are the same kind, that fusions occurred, telomere, analogous to the human condition in them as well. What are you doing, Jeff? What are you doing? You are giving me heartburn. I had to do it to him though, I didn't have a choice. I simply had to come after him for this because he's just on my screen and he's saying incorrect stuff. Okay, let's continue.
And so they're not designed to just fuse. They're not just hanging out there waiting to fuse.
As far as the genes inside, though you said it was inside a gene, do evolutionists think this is a pseudogene that it's inside? And also, what is a pseudogene? You talked about that in your book.
Yeah, so most of the pseudogenes, the so-called pseudogenes, are long non-coding RNA in the genome. But most of them now that they have analyzed are functional and important to human health, of health defects in humans. So actually, pseudogenes, or more properly, long non-coding RNA genes.
Okay, you guys, he's going to talk about junk DNA, and I don't care about junk DNA. And the reason I don't care about junk DNA is because one very simple thing disproves the notion that, and I do mean this, like rarely do you get to say that something like disproves something, but I do feel confident to say that this does in fact disprove the notion that the entire genome is functional, right? Because creationists need the entire genome to be fully functional for their perfect creation genetic entropy stuff. So is the genome fully functional? No, it is not. And the way that we know that it's not fully functional is because of knockout tests. You can knock out megabase portions of a mouse's genome, just get rid of them, knock them out, and they can reproduce and have offspring just fine. And then their offspring can also have offspring. So like if the entire genome was functional and necessary, you wouldn't be able to run any kind of knockout test without some kind of actual consequence, more likely lethal consequence. Fruity Lurlips just asked, what is my shirt? My shirt says, actually, let me just show you what my shirt says. I actually quite like this shirt, and I like sharing my shirts with you guys. It says, find what you love and let it kill you. And this is how I feel. I felt this was appropriate for today's topic of conversation. Find what you love and let it kill you. Watching Jeffrey Tompkins like content actively is bad for my cells. It makes them more likely to fail in division. they become potentially cancerous when I'm watching Jeffrey Tompkins's content. Okay, so we don't need to talk about junk DNA. We don't need to talk about pseudogenes. We know that the entire genome is not functional. Moving on.
Translated into a protein. I do suspect that a lot of those regions were not represented in their analyses.
Awesome. This discussion has been a huge event going on YouTube, and people have been talking about your method. People's been talking about your chromosome, the alleged chromosome 2 fusion claimed by evolutionists, and there's people on both sides. They're debating the subject back and forth.
By people, he means me. I'm the one.
And so it's been a very big controversy there. And when you think that that the biggest thing that the evolutionists would criticize and say, no, we're not doing that, was all the cherry picking that you've accused them of. But instead, what they've done is latched on to your particular methodology, where they challenge whether or not if your method can get this correct number. So we're going to deal with some criticisms. The first one is what I like to call the math argument, because it's one where, well, before I do that, I need to clarify something. The gap and ungap. That means with including indels, which are insertion and deletions, or without insertion and deletions. One of the criticisms was on your first paper didn't include.
Well, no, actually, Apologetics 101. That is not actually what the gapped versus ungapped comparison is. And I know I told you the difference to this when you were in my live chat. We're going to be addressing Apologetics 101 quite a bit moving forward here, because Apologetics 101 was in the chat for the Tompkins, for my Jeffrey Tompkins video. He asked plenty of good questions. The questions were good questions, but the problem is that he went into this interview, and I feel comfortable saying this, and I feel that I'm going to support this notion here momentarily, without really understanding what the issues were for Jeff and without really appreciating what I did. Okay. And I feel like I'm going to be able to support this moving forward. But that's going to be my thesis statement with regard to our interviewer here, Apologetics 101. So what he's saying to Jeff here is that he's saying Erica has an issue with your methods, right? And I do have an issue. I'll sum it up for you guys here right now. From 2011 to 2015, so it's 2011, 2013, and 2015 papers, Jeffrey Tompkins uses an ungapped method. Ungapped methods cannot handle small differences in the genome and immediately stop the alignment the second it encounters an issue. Because of this, and in addition to I believe it's the max HSPS parameter, the ungapped does something funky with how it returns the best hits as well. You end up with non-100% identities of an entity to itself, right? You can't get a 100% identity of something to itself when you use ungapped parameters. That's why they don't work. Now, that's why Tompkins's parameters don't work. Now, his 2016 and 2018 methods, he doesn't weight his data. So we already talked about those issues earlier. So the issue is gapped versus ungapped, weighted versus weighted. And then as a side note, methodologically speaking, you do have to take into account how you're going to deal with indels. Is it going to be one indel is one difference, or is it one indel is, let's say it's 1,000 base pair insertion? Is that one difference, or is it 1,000 difference? All of those things are going to have an impact on what your return is, like what you actually get as your percent similarity. But only 2 of those are catastrophically lethal to your methods, and that's gapped versus ungapped and weighted versus unweighted. So knowing that, now that you guys know that, let's move forward.
Include gaps. Is that correct? Or the other?
There was an initial paper I did, I think back in 2013, where I did not include gapping in the algorithm. And it turns out the algorithm was bad anyways, because it was kicking out sequences in large data sets.
Okay, Jeff, I need you to be upfront here, because the reason why you redid the 2013 paper in 2015 was because of the bugged blast version, but you performed an identical analysis with ungapped as your parameters. So your problem in 2013 was a bug, but your problem in 2015, let me rephrase this. Your problem in 2013 was a bug and ungapped. Your problem in 2015 was the bug was fixed, but you still used ungapped.
And I actually talked about that in my later paper. So it's not like I was, not discussing the issues that were going on later on. So yeah, people that want to be naysayers and so forth will go after something you did originally that, and not acknowledge the most recent work you did. And fundamentally, people have presuppositions.
Okay, so this is why in my video I systematically cover all of the papers on this topic. So yes, we do cover your errors in using ungapped early on and why those are errors. And the way we showed that they were errors is we compared something to itself. Then later on, we talked about your 2016 methods and your 2018 methods and why those were also flawed. So we covered both. I'm not being a naysayer because of the bugged algorithm. I'm being a naysayer because you're bad at mammalian genetics.
Some people are bound and determined to deny there is a God. They don't care what evidence you give them whatsoever, and then they want to nitpick something that really doesn't. It's kind of an esoteric angle that really doesn't invalidate what you really discovered later on.
It's absolutely not esoteric. The gapped versus ungapped parameter is critical for how he runs his analysis. And like I said, it also messes around with how the best hits are returned. Like I'm not exactly sure how that happens, right? Like even ungapped, you should get theoretically 100% similarity between something and itself. But using Jeff's methods, you don't. And I double checked that I'm using his methods. I copied and pasted his methods directly into my configuration file, like verbatim. It is a copy and paste of his parameters, right? And I also got the same similarity that he got for humans and chimps using his methods. So I've got a decent control here for that. It's not what I'm doing. It's his methods that are problematic. And I did all of this on camera, remember. So both, at least Apologetics 101, who was in my live chat, saw that I used Jeff's methods, or at least he claims that he saw that. He claims that he watched the whole video. And I presume he did because he was there for the whole chat. But moving on.
Anyways, in fact, there were problems with that algorithm. I reported those to the NIH, who created the algorithm, and I was actually corresponding with the programmers there at the NIH and sending them data sets because I was trying to convince them, hey, your algorithm is messed up.
This is not cool of Jeff to say, I don't think, because Glenn Williamson was the one who brought the error to Jeff's attention. And if memory serves from their email exchange that I saw, Jeff was like doubtful that there was a bug at first. He was convinced that there was a bug, and then he pestered the NIH, right? But that was not how that initially started. So I don't think Jeff can take credit for discovering the bug, to be honest.
Kicking out data. And I finally was able to do that. I actually had to send them data. I had to send them results from analyses, and they did fix the algorithm. And so in later papers, I did use gapping, and the algorithm was behaving itself and was not kicking out data. And so, yeah.
So your initial
people will focus, on an earlier paper, and then you produce research later on, and they ignore it. So,
right.
I mean, what can you do?
Right. So your initial.
Okay. But we didn't ignore it. Just reminding everybody, we didn't ignore his later work. His later work is also bad for different reasons.
The matter. Yeah, go ahead.
I was going to say, initially, some earlier ones, it was ungapped because of the fact that you had a bug in that particular algorithm, but then that problem got fixed and resolved.
Well, no, I was using an ungapped algorithm because actually a lot of evolutionists were using ungapped algorithms in the early days as well.
Yeah, in the early days, like early days, we were using ungapped because we were using highly similar pieces of DNA. We're comparing highly similar chunks of DNA one to the other. So yes, like it was appropriate at that, probably, honestly, to be honest with you, it probably wasn't even appropriate then. But you'll notice that no method uses ungapped now, nor have they been using ungapped like since, I don't know, the late 90s, something along those lines. So, I mean, this is like in classic creationist fashion, we're using methods that are outdated by decades. But no, like, so ungapped is not an appropriate way to do it. You saying that secular scientists did this early on it would be similar to me saying that, like, if I bleed someone of their yellow humors or whatever, it's fine because doctors did that early on. It was wrong when they did it early on. That's why they don't do it now. I don't know if you can bleed someone of their yellow humor. Pretend I made a better example.
Okay.
So actually, this particular algorithm has been, greatly improved over the years, and so you just got to use the tools that you have at the time. So, yeah, the most recent paper I did, I used the most liberal, gapping that I could on the algorithm, and yeah, people will throw out anything to discredit you. It doesn't matter what it is. So,
right. Some people say that they say, yeah, but even though he switched back to gap, it was unweighted. What does that mean exactly when they say that they don't think you weighted your sequence?
Takes into account the weighting.
You
can't set the weight really of the algorithm. You can statistically you can set the gap extension.
No, the algorithm, you can program an algorithm to weight or unweight for you if you want, but Jeff, you didn't program it to weight. You didn't. You simply did not program it to weight, as evidenced by us playing around with your published CSV earlier. You didn't program it to weight, and so you got an erroneous result, and that is an incorrect way of coming to a conclusion with regard to various sequences of various similarities. You have to weight it. Like, mathematically, this is what you do. I don't know how to help him with this.
I don't know what people are talking about. I mean, it's non.
Oh, wait, I'm sorry.
Extension, you know. I don't know what people are talking about. I mean, it's nonsense. I did what I could to create, you know, a situation where the algorithm could get over at least the small gaps. The algorithm can't get over the big gaps.
This doesn't have to do with anything. You didn't weight the data. This doesn't have anything to do with your liberal gapping parameters or whatever. You didn't weight the data in 2016 and in 2018. So like there's been a bait and switch here. So first he talks about, oh, when I used ungapped, it was appropriate, except no, it wasn't. Not when you used ungapped, it wasn't appropriate. And then he's like, well, you know, then I didn't do ungapped because of a bugged parameter. There was a bugged issue in BLAST. It's like, no, but then you did it again once the bug was fixed in 2016. And he's like, well, but then I didn't use gap or ungapped anymore. I used a gapped parameter. Yeah, and then you didn't weight it. And when you do weight it with your gapped parameters, you get 96% as the similarity. What do you do with this? Again, like how does he reason through this? I don't understand.
Yeah, why aren't people talking about, oh, why could he only align the 30% of these 30,000 base chunks of DNA with the most liberal gapping he could? And no one's talking about, you know, the 2/3 of the DNA sequence that the algorithm, you know, couldn't even align because the difference got so extreme, the algorithm had to break off its alignment.
Does this
why
affect
aren't you guys?
Does this affect your number any? Whether it's weighted or unweighted?
Like I said, the weighting is included in the setting of the parameters of the algorithm.
No, it's not. No, it is not. Like your published CSV shows us mathematically that it is not. It's not included in the algorithm. You guys saw that for yourselves.
So in other words, it's automatically set within the algorithm itself.
Yeah, you set the statistical threshold, you set the gapping parameter. It's like I say, people don't care what you discover. I mean, I could discover 50% DNA similarity or whatever. It doesn't matter.
I mean, that's just not true. Like, I'm struggling here, you guys. I'm struggling. You know, I try to assume best intent, but I am struggling with this. I am very much not sure how he can just sit there and say, like, either he doesn't understand what's being asked of him, because it sounds like he's talking about gapping and gapping parameters there. That's not the problem with the 2016 and 2018. The problem with those latter two is the weighting. But then he's saying the algorithm weights it for, well, okay, but it did it, not in what you published, right? Why is it that when we take your data set and we average up the percent identities, regardless of the weighting, right, which they're not weighted clearly because it's just a raw p-ident, why is it that we get your published number for the human chimp similarity when we average those, and we get the conventional number when we manually weight them in your published data? Help me out here, guys. I really want to assume good faith. I really do.
And you know, unless the Holy Spirit is really working on that person to reveal the truth to them, apprehend the truth. And I would suggest to people out there, you know, you do what I did before I became a Christian. I was driving down the road in my car and I said, God, if you are out there, please do something. Reveal yourself to me. You know, I'm tired of living life the way I am.
I'm gonna show a clip. Now, I think they're trying to think that some people will try to take all your research in a particular paper. They say that they're you know, like, and put it in, and they put the numbers in an Excel type document.
That's his data, Apologetics 101. His data, when you it's a CSV. It goes into an Excel spreadsheet. Some people, oh God, oh, okay. I mean, okay, I'm going to get a little mad. I got a little mad when I listened to this the first time, and I'm probably going to get a little mad again. I'm probably not going to be able to help myself because there's some stuff in here that's like insulting to my intelligence and my capabilities. And like, I do my best to be thorough. I ran this by other people, right? Like I didn't just do this in isolation. And I just don't really know what to do with this. Apologetics 101, like I feel like you didn't do your due diligence before this interview, despite the fact that you told me in the comments section that you watched my video to the point that you had it memorized. Okay, Let it play.
And then they'll set
I can tell you what people are. I can tell you what people are talking about. So there is a guy in Australia. I don't know if he's still alive. He got cancer actually a couple years ago. He is a computer programmer, and he literally attacks creationists. It doesn't matter whether they're talking about human chimp DNA or whatever. So anyways, this guy's a computer programmer, and him and some other guys were working for a financial trading company, and they started their own company and became millionaires literally overnight. Thinking about all this money he made and how he was going to spend his life now attacking me and other creationists. So this is the guy that put out that video, and it's utterly bogus from start to finish. And yeah, it's I know what you're talking about.
We're speaking of
he's a nefarious. He's a nefarious character, basically.
A nefarious character. Oh, Glenn, oh no, you're a nefarious character. I guess I'm a nefarious character too, since I double-checked your methods. What a victim complex you have to have. Like, oh my God, like the fact that someone double-checked my methods and did it in a sassy way on YouTube means that they are a nefarious character. The way he phrases this makes it sound like he's like an agent of the devil or something like that. Like, what? That's so pitiable. That's such a pitiable attitude to have. Glenn just double checked you, dude. And like, I think this deserves a little hop, skip, and a jump over to that video that Glenn created that Jeff commented on. It's called Tompkins Methods Are Utter Poo. So you can understand why he got a little bit mad about that. But it doesn't really matter because like Jeff is just objectively wrong in a mathematical way. And like, I want you guys out there who are watching this, dude, double check me, right? Double check me and Glenn on this. And you let us know, like because Glenn did the same thing well before I did when we looked at the published CSV. You guys check it out and let us know. Like, was that an incorrect criticism to make? You have to weight your sequences. Okay, so here is Glenn's video. We went over it in my Tompkins video, but here's what he did the same. I did the same thing as him here, right? Where I took the data, I put it into an Excel table, right? It's already in CSV, Apologetics 101. So we just put it into a table so it's easier to digest. And you can see that I basically did the same thing as he did. And you can see down here, this is where Jeff comments because Jeff was so upset about what Glenn had published here. And Jeff's comments here are basically like, Glenn's sample size is small and mine is large because Glenn shows that weighting is important by giving an example using fuel efficiency, right? So at the very beginning, he talks about, or actually it's over here. He says, if I take my car out twice last week, Sydney to Melbourne, right? And you have, I got busted for saying Melbourne. So Melbourne, sorry, Australians. So Sydney to Melbourne, fuel efficiency there. His house to the supermarket the next day. What's the average fuel efficiency, right? You can't just average those two locations because the fuel efficiency is going to be different depending on how long you're driving. So you have to weight it by the distance, excuse me. And Jeffrey Tomkins, geneticist, PhD from Clemson, he thinks that the problem with that analogy is that the sample size in Glenn's example is n equals 2. It does not matter what your sample size is. Weighting is what you do methodologically here. The sample size doesn't matter. You could have, he's like, I calculated an average using a sample size of 18,000. It doesn't matter because the sequences are different lengths. In all 18,000 sequences, you have different lengths. Maybe not all of them, but in a lot of them, the lengths differ. So you can't just brutally average them. Guys, is this not middle school? Tell me in the chat right now, when did you learn, at what age were you taught in your education to weight things? When did you learn this? For me, it was middle school. So we're going to bop back over to our pal Jeff. He's going to tell us exactly what he complained about there in the comments.
I know who you're talking about, Glenn Williamson, right?
Glenn Williamson, yeah. He calls himself Ruhif, and I'm not the only one he's attacked. He will literally use email addresses in the creation science. He's attacked multiple people in our ministry, at other ministries. Even if you're just an intelligent design person, you don't even necessarily believe in a biblical timeline or whatever, he will figure out who you are and attack you one way or another. But one interesting point he made at the very beginning.
This is so pathetic, Jeff. Like attacking you, you're getting peer reviewed. Like you won't publish your results in conventional journals. You publish them in a creationist magazine. right? So you're getting peer reviewed by people who are going out of their way to check your work, and you're calling that being attacked. That's not an attack. That's peer review. That's how science works. Now, if you don't like that Glenn swears or that he's sassy to you, you're allowed to have that preference. But what you're not allowed to do is call it like a baseless attack without first justifying why you used the methods that you used, which you cannot do.
Of his video is he said, oh, well, Tompkins just determined the average of these DNA similarities for all these alignments. And he said, here's an example. Let's say you drove here and it was only, I forget what he said, maybe 20 miles, but then you drove to this other part of Australia and it was 1,000 miles. And then you took the average. Well, what's that? That's a sample size of 2. What was my sample size? 18,000, you know? And so statistically, his argument was nonsense. And then
absolutely not. Absolutely not, because you have highly disparate small sequences compared with highly similar enormous sequences. You cannot just average them. That is not going to, statistics isn't going to magic away your blunder.
And he never explained all of the sequences that had super low similarities. And he also only looked at the alignment similarities, ignored the 2/3 of the sequence that didn't even align. And so my paper was totally accurate. The average identity of those 18,000, the mean of those 18,000 sequences was 84%.
My paper was totally accurate, you guys. This is what I'm going to start doing when I submit a manuscript. When I submit to a journal, I'm just going to include, like, listen, you guys, my paper is totally accurate. Don't peer review me. Don't attack me because my paper is totally accurate. Okay? Don't worry about it. You don't even need to read it. Like, he calls himself a scientist, right? I mean, and here's the thing. I know Jeff does okay work. Well, I mean, I think he does okay work. I'm not a plant geneticist, so I really don't know anything about plants in general. But he has published in conventional journals about, like, plants. It seems fine. He's not the sole author on them. And then, of course, he turns around and botches mammal genetics like a pro. It's the weirdest thing you've ever seen. I mean, it's got to be some of the most intense cognitive dissonance out there, the stuff that Jeff Tomkins is doing, because there's just no way that you can publish. I mean, I think he's been in conventional journals like as first author like 5 or 6 times even. He's been in a lot of them, but as first author, he's even got like 5 or 6 papers. A guy who's capable of doing that, I don't think is capable of not understanding why you need to weight your sequences by length. I just don't think it's possible.
And so he went from all these weird, stupid angles. Some of them were just totally bogus. And so, yeah, that's the video that's out there. And anyways, so I'm not the only target of this guy.
One of the things I found out,
other people have been attacked as well.
The target attacked me when I'm sitting in my basement with my battle plans mapped on my big, like, medieval, like, stone desk as I'm charting my line of attack against Jeffrey Tompkins and fellow young Earth creationists. I'm moving my my pawns along the makeshift, like, cartographer's map of how I'm going to lead my evolutionist army to victory against these. Get over yourself. Come on.
Yeah, one of the things I found
What's
out
that?
is that all of these critics seem to be bouncing off of each other, like they're using each other's arguments, and they're just kind of almost absorbing each other. I know that like, you know, you have Williamson or Williamson, the Glenn Williamson, and he said he got his information from Dr. Joshua Swamidass, and
we love Josh Swamidass. We stand a cool theistic evolutionist on this channel.
And Erica.
Well, Swamidass is another. Yeah, let me mention Swamidass.
Right.
So basically, this one study that I did, it literally took 6 months of time on 2 servers, each one having 40 logical cores and 384 gigabytes of RAM. Last study that I did, and Swamidass claims he went home from work one night and repeated my study in a couple hours. I'm like, are you kidding me? You're a flat.
Hold on, you need to hear him say this.
Work one night and repeated my study in a couple hours. I'm like, are you kidding me? You're a flat out liar.
I hope Josh sees this, because Josh characterized Jeff as a nice enough guy. So there you go. I don't think Jeffrey Tompkins is a nice guy. And frankly, I have opinions about how honest he is as well. Now, Swamidass didn't claim to replicate his entire study. So let's pull that up. Mind the controls. let's just we do a check. Okay. Pull it up, pull it up, pull it up, up, up. Yeah, true, Tarleton lives. I am so smart. Nobody can compare to me, and they are lying if they don't think I'm a genius. True. Direwolf 96A, Hovind or Tompkins. At least Hovind is funny. I mean, Tompkins is just he's kind of boring, honestly, as an individual to watch. Of course, to my understanding, Jeffrey Tompkins has not been at least once found guilty or at least accused of domestic assault, structuring, and harboring very evil people that their title will get me demonetized. Okay, so here is Peaceful Science. Here's the article that we're talking about here, right? This is Human and Chimp Similarity, Mind the Controls. So Swambados responded to this article quoted to me by GES. There it is, the analysis of 101 trace chimpanzee read data set. So this is his 2016 paper, and I know all the paper titles by heart because I read them all, unlike Apologetics 101, who's going to insult me later on, and it's really irritating. I once wrote about this elsewhere. He talks about it here. Human-chimp similarity, scientific evidence by Logos Forum. Very familiar with Tompkins' work. He seems like a nice guy, but his study is deeply flawed. The data is freely available, so I reproduced his work for fun. He's not saying he reproduced the entire study. He's saying he reproduced his methods, right? That's what I did as well. I found he made some large errors by excluding some basic controls. Including controls, we find human-chimp similarity reads is about 98%. For comparison, similarity of rats and mice is about 80%. Rats and mice are the same kind. Seems that humans and chimps are too by using Tompkins' methods. And then he basically does several controls. He said there are many ways to do it. Some are better than others. Exact number isn't important. The key thing is to lay out a number alongside controls. It is a relationship between the computed similarity that controls and allows us to interpret it. Tompkins' proposal is reasonable. He suggests measuring similarity between chimp reads, which are short fragments of raw data, and the assembled human genome to eliminate bias introduced by using the human genome to scaffold the chimp genome. This is actually a clever idea. Let's use it. So he takes the chimp reads and computes the average similarity between these reads and the reference genome. He computes 85% similarity. I agree that that's what he's got because I got the same number as him. So Tompkins is saying that Swambados is a liar. He's calling him a liar for saying that he reproduced his results. He's not saying he did your whole study, Jeff. He's saying that he reproduced your methods, like what you did, your work. And I think this is good support right here. You got the same number as you using your parameters. That's exactly what happened with me as well, and why I feel confident that I interpreted, that my BLAST program was capable of interpreting the copy and pasted methods of Jeffrey Tompkins correctly. I agree that's what he got. I got the same number as him, but how much is because error in the reads? There are no controls, so we don't really know. So he basically just tries a chimp to a chimp, and he gets 87% using Jeff's numbers. Then he tries a human to a human and gets 89% using Jeff's methods. Then he tries the human to the chimp, so the reverse of the initial one, and he gets 87%. So there's an error in the methods, right? He's using ungapped, and I believe he doesn't use max HSPS or max target sequence. I'm not sure which one. I'm going to pull it up in a second. Someone was also asking about that in the chat. How do you not get 100% with ungapped? And I'll explain in a moment. At least I'll explain what I think is going on. So this is what he's accusing Swambados of being a liar on. It's like, it's not Swambados's fault that your parameters don't work. And like, what Jeff is doing is that and he'll talk about this later, he starts these really long analyses. He boots up his computer, starts an analysis, and then it runs forever. I'm doing the same thing and have done the same thing for this type of analysis. I'll stop sharing the screen for a second. You can see over there, right? That's my other computer where I'm currently running BLAST. And in currently running BLAST, I'm running a 30 million base pair comparison of chimp 2A to human 2. I'm rerunning something because I botched it the first time. So I'm rerunning that. And I started that run last night and it's still running, right? So you start it and you leave it running. That's great. So Jeff starts these big things. He leaves them running. And then he's like, how could anybody reproduce my work? My brother in Christ, all Swambados did and all I did is took a smaller number of base pairs. Your work should be able to reproduce a 100% identity, or should be able to produce a 100% identity between two identical things, regardless of the number of base pairs. And it doesn't. That's the problem. That's the issue with Swambados's, with a lot of Swambados's, There's an issue with a lot of Tompkins' methods. Okay, back to it.
It's just nonsense. And I wished I'd have saved that quote. He put it up on his blog, and I'm thinking, that's just a bold-faced lie. You didn't go home and one night on your little PC and repeat a study that took me, 6 months to do on these powerful servers. It's nonsense. And so, yeah, and I think Swami Das, and who's supposed to be a Christian, and there's Glenn Williamson, who's obviously a complete and total pagan.
This guy, he's so mad, right? Like, he's so angry that anybody dared to test his methods. Again, Jeff, it's not everybody else's fault that you ran an incorrect analysis that flunks middle school math. That's not everybody else's fault. And like, this really makes me mad because Joshua Swamidass is incredibly kind and assumes the best intentions in everybody, even Jeffrey Tomkins. And Tompkins turns around and calls him a bold-faced liar. Super uncool.
Why are these guys collaborating?
Yeah, and there was a video.
Because it's not team sports, Jeff. This is science. You don't have to pick it's not like a team where you're not allowed to work with somebody who you disagree with on. In science, people collaborate all the time with folks who they differ on religious views with. Because get this, if you're a good scientist, your religion doesn't impact your science. So it doesn't matter if you differ on religion with people, right? Only younger creationists let their religion get in the way of their science. Only them.
Just posted a few days ago by a evolutionist named Erica from Gutsick Gibbon, and she actually posted a video where she was trying to target that, and she used a lot of these same arguments. She used the
Yes, I absolutely did. I ran all my own analyses for his 2011, 2013, and 2015 papers. That's all me. I did all that on my own time. Now, Glenn helped me a lot with figuring out how to use BLAST. And get all my stuff situated. My husband helped a lot as well, but like, that's all my own work. But like, what else would I do? Right? Like, what else would I need to do to Jeff's 2016 and 2018 methods when like they're proven wrong by downloading his own files and just showing that he doesn't weight them? Like, there's nothing else that needs to be done here. Why reinvent the wheel? And also, like, I'm a scientist. I like citing my sources.
The arguments from math, and she used the arguments from she focused on the bug in the original system or whatever the case may be as an algorithm, and she said she'd done these tests, these experiments, and she also had her regular computer system. She said she used BLAST. She said, and she said that she did a study on the first one was on the things that we would agree is the same kind of animal. And she said that she come up with all these differences and stuff like that. Now, of course, that's easily explainable. I mean, we don't expect them to be exactly alike anyhow. But then she also said that she also tested it against itself, like using your method. And she said that she didn't get 100% identity.
Okay. So pause here. We need to be clear what we're talking about here. The test that I did, again, on my own computer, on my own BLAST, those are for the 2011 to 2015 methods. The ones that I relied on other work that had already been done for was the 2016 to 2018 methods. And specifically, it isn't that the kinds were different. I'm not saying, this is what I'm saying, that Apologetics 101 doesn't understand what I did and why it was important. A line and a link should not be 100% identical under anybody's model, under conventional science or under young earth creationism. No one is saying that. But they should be more similar than humans and chimps. Or they should be more similar than a human and themselves. Or they should be less similar, than a human and themselves, right? But they're not in either of those cases, right? Because the way that Tompkins is running BLAST is bugged, or not bugged. It was bugged, but it's also not correct. And I'm going to show you guys another key thing that he left out that I think heavily influenced his results here in a moment. We're just going to run a little bit of Tompkins stuff here on camera. I think that would be fun, don't you?
Now, of course, Swami Das said the same thing on his Peaceful Science blog, like you mentioned, where he tried to test rats and mice, he claimed. Again, to me, I'm already automatically skeptical of this because they're using their personal computer. Granted, they're using some similar algorithms, but they're using the personal computer. She's claiming that she did it exactly as it's spelled out in the paper, is what she's claiming. But I read the methods in some of your papers, and they're pretty advanced. So Apologetics 101 does not understand that there's a difference between the equipment that you're using and the parameters in the program. I am using the exact same program, and the program is only influenced by the actual type of computer that you're using insofar as how fast it runs.
The program is identical. BLAST is going to operate the same way on my podunk little computer as it is on the best supercomputer in the world, but the supercomputer is going to go a lot faster. The rest is the parameters are what's important. So how about you guys, we do that again? And I hope you appreciate he's called this home experiments in quotations at the bottom, which is super like condescending, I would say. I can't remember if that comes from Tompkins or if it comes from our friend here. So let's do a few things here. I'm going to share my screen here brutally. We're going to share the screen. We're just going to do an entire screen share. Click, share. Okay, so here is the, let me fix this over here. Where's my other Excel? There it is, config. Okay, so you guys can hopefully see everything going on here. I'll check in a second to make sure you actually could see everything. But for now, we are going to adjust these windows, and we're just gonna have to show Apologetics 101 again, that my methods are Jeff's. Verbatim. Okay, so this section here, good. This is like based off of the default parameters that you want to use in BLAST. And we'll notice that like we got our output format there, but the applications to it it's gapped is automatic, but it's also max HSPS 1 and max target seqs 1, max target sequences 1. So here's Jeff here. We've gone to parameters, Control F parameters, and his parameters are right around here somewhere. Is that it? Yeah. BLASTn parameters utilized were as follows. E-value 10, word size 11, max target sequence 1, dust, no soft masking, false, ungapped, number of threads 6. And hopefully you can see down here where my cursor is, the exact same format. E-value 10, right there. Word size 11, right there. Max target sequence 1, dust, no, soft masking, false, ungapped. And then if you scroll to the side, you can see number of threads, or actually I didn't do number of threads 6. That's how fast it runs on your computer, but mine doesn't have that many threads. So I actually can't run 6 threads at once, but Tompkins can because he has a big computer. So these are the parameters that actually influence what's going to, how BLAST is going to run the algorithm. They are verbatim, directly from Tompkins. So now that we have that in mind, what do you guys say we run something? Let's just run a little something here. Let's pull open our guy here. Hopefully, let me make sure actually that you guys can actually see this. Please, God, let you guys have been able to see this whole time. Necess, yeah, okay, you can see. If you know that I play Necess, then you can see. Great. Okay, so here's our system, right? We're gotta change directories to get to the right place, change directory document, change directory genomes. Okay, so now I initiate how I actually run this. Generate random win. This is the executable or whatever that I utilize to basically, what's the word I'm looking for? It's like streamline. It streamlines the process. My husband made it. He's a computer programmer, and he was basically like, okay, well, doing it this way, we can basically kind of package it into a single executable so that you don't have to have different code in a different place that you have to execute at different times to create the random sequences and then actually do the alignment. And you don't have to like narrow it down by chromosome. You can download full genomes and do it that way. So here's Dogful. Dogful 1, this is a downloadable Canis familiaris. It's just the dog. It's a downloaded dog genome. And directly underneath it is Dogful B, a copy of that dog. This is the same dog. It's the same reference genome. It's the same individual. So I'll click enter for dog 1, and we'll compare it to dog 2 here. Okay, so there's a lot of different variants. That's for like a top level. I believe this is top level downloaded, so it has a lot of different variants. But this is the critical portion down here. Here's our chromosomes. So we're going to compare chromosome 1 to chromosome 1. And here's what we're going to do. 100 sequences. 100 sequences. Enter. And we're going to use the good method. So it's going to take a couple of seconds because this is like a super fast way of, it's like a pretty efficient way of running. And there you go. Max target sequence 1, dog to dog, chromosome 1 to chromosome 1, the same sequence, 100% similarity. Good to go. And my husband was even cool enough to put weighted and unweighted, like he programmed this in. That's how we know that BLAST doesn't do it automatically because he had to program this in. So There we go. So now let's try Jeff's method. Generate random win. Same thing, dog to dog. We'll use chromosome 1 to chromosome 1. We'll generate 100 sequences, and BLAST is like the program that he's generated here, like the code that he wrote, the script, is smart enough to know that since we've just done 100 random sequences, it's going to use the same previous ones. So see, it knows we don't need to make them. And this time we're going to use Tompkins 2013. So there you can see, it's his exact methods minus the number of threads because my computer is weaker. E-value 10, word size 11, max target sequence 1, dust no, soft masking false, and ungapped. Okay, so it takes a little longer to run Tompkins' method because he doesn't have a very critical parameter in there. And while that's running, I'm going to show you what that parameter is using Excel here. So remember that with good that returned 100% similarity in max target sequence 1 here. What we did is we had this little parameter here, max HSPS 1, right? Max HSPS 1. Tompkins' 2013 methods, they don't have that. And my understanding of how BLAST works is that's what's making sure that we're only returning like the best matches instead of like any willy-nilly partial 10 base pair match. And that's what drags his percent similarity down, despite the fact that he's using ungapped, or that's what pulls the similarity down further with ungapped, even when you're comparing something to itself. So the ungapped is the problem. The fact that he's missing this, I guess, critical parameter here makes the problem worse. Really, it's mostly ungapped. And there you go. There's your percent similarity for Tompkins' 2013 methods. 89.1% weighted similarity of that dog's chromosome to itself. You don't need to compare the whole chromosome here, Apologetics 101. The fact that it can't return an identical similarity for the same 100 base pairs to like in one to the same organism, right? That it can't find that 100% identity means the methods are flawed. And that's the long and short of it, you guys. That's why Tompkins' methods don't work. So I'm going to stop sharing the screen there so you guys can hopefully appreciate the problem here. Now, I know what you're thinking. You're thinking to yourself, what if you did that same exact comparison, but you used just 2016 and 2018 methods? You're going to get 100% because the problem isn't the parameters, it's the weighting for those 2016 and 2018 methods. So hopefully Apologetics 101 understands it now. But let's flip back over and hear more from Jefferini over here.
Well, I'm sitting there thinking, okay, it's kind of hard for me to believe that they're set up exactly in a laboratory situation where they will be able to actually cover this in there. I know if she's watching this, which she will be, she'll probably get offended. Oh yeah, I did it exactly as it was spelled out in the paper. But she's not. Again, it's the whole. I don't know if she's set up with all the right equipment, all the right gear, and even whether she even did your method perfectly, regardless.
Literally, it does matter that I did his method perfectly, and as you saw, it is verbatim, copy pasted even in the same order from the paper with the absence of number of threads, which has to do with my system's abilities. But my system can run BLAST. BLAST doesn't work differently on a master computer as it does on a regular old podunk, regular little monitor or whatever, a little laptop. That's what I meant to say, little laptop. Those two things are not going to differ at all. It's going to differ in how fast it can go and the capabilities as far as like how many threads, how many times can it run it at once. That's where the difference is going to be. I don't know what else to say. And you know what? While we're listening to this in the background, I'm just gonna add to my configuration number of thread 6 to my Tompkins configuration just to shut everybody up who might say the number of threads, they don't understand what it is, but they're like the number of threads is gonna influence it somehow. So we'll let this continue to play.
Which she said she did. But in any given case, how would you respond to people saying that that they've used this experiment, that they've tested it on similar kinds, that they've tested against itself, the same kind, not just the same kind of animal, but like test my DNA with my DNA, and they say that okay, they pounce on it, they say nope, the method doesn't work. I mean, how do you respond to people like that wants to claim that they've done these types of experiments? Now, granted, like you said, it took you a while to actually do the experiments, and you got resources, you got much more powerful stuff. But how do you respond to people who said that they basically try to replicate this back at home and they say your methodology doesn't work?
Well, right off the top of my head, I don't believe them because I would basically log in to a Linux server and start these jobs with shell scripts running and then log out, and the jobs would just keep running for weeks because the data sets were so big. And so I know Swami Das, what he said was utter nonsense when I first read that. And I almost felt like sending the guy an email, but I didn't, calling him out on it because it was so ridiculous. I have no idea what this Gutsick Gibbon girl did. But yeah, you're right. They just kind of feed off of each other and put out all this stuff on the internet.
Okay, so what was wrong with it then, Jeff? Like, their whole argument here is you didn't run the experiment using my powerful computers, and instead you ran like a smaller subset of the data. So my methods are fine. No, the problem is that your methods can't even return an accurate result with the tiniest data set imaginable. That's the problem, Jeff. That's how you know that you didn't utilize controls. And for you to call Swamidass a liar about that, when you evidently don't even know what he did, because you can't sit here and say, here's the reason why I know Swamidass was incorrect. His whole thing is Swamidass said he did my whole experiment, but he couldn't have possibly because it took me so long. Well, he doesn't need to. He could run a control just like I did, a tiny data set that shows that your methods don't work. And then it doesn't matter what your size of data set is. It literally doesn't matter. If they can't work with a small data set, then you shouldn't be applying them to a large one. You simply shouldn't. So let him continue.
Seriously doubt any of them have come close to replicating what I did, because I know the amount of time and effort and processing power it took. And to think these people went home and did it at their house is crazy, unless they had access to really powerful servers like I do. So right off the top of my head, I know Swami Das that was utter nonsense what he said, and yeah, and I know that Glenn Williamson, the stuff he said was very misleading and very caustic, and but I don't have time to.
Really, Jeff, it was misleading. What was misleading about it? Tell us when you get an opportunity, whenever you can, or you could have done it in that comment that you chose not to do it in. Tell us why the sample size means you don't have to weight it. Tell us statistically how that works. Since you're appealing to statistics, how does that work? Let us know. Let us know while I'm doing this.
I feel like sometimes I'm in a whack-a-mole type situation, where someone pops up and says this and that, and even though it's just utter nonsense, someone else is popping up over there and someone over there and someone over there. And before I know it, I'm spending all my time trying to respond, to people that are creating all these diversions when I could be actually doing real research instead of, doing this whack-a-mole kind of thing. So
I don't like that.
I only have so much time in the day, and like I say, there's going to be people out there that are so caustic and anti-God that they'll do and say anything. Like
it's anti-God, right? All these other people, other Christians who profess and work at places like BioLogos, but they are bona fide Christians. Because they accept conventional science, they must hate God. That's it, right, Jeff? That's it. Golly, he's just so. I just don't like it. I don't like Jeff. He's a jerk. He's acting like a jerk here. He's like calling all these other people liars and saying that they must hate God because they found results that run contra to what he's found. But at the same time, he doesn't want to talk about why they're wrong, right? He's like, oh, Swamidass, he didn't do it the way I did it, or he ran a smaller version of it, so therefore I won't even look at his results. You know what Tompkins could do here, you guys? This is all he needs to do. Run what Swamidass ran. Just prove him wrong. Just run what he ran and do it in your big, bad, cool experiment. This was back in 2016, so I know you're not going to do this. It's been almost a decade, but that would be a really easy way for him to prove all these haters wrong. Just run any kind of control. Any kind. It's always human and chimp, human and chimp, human and chimp. Nothing else to contextualize those numbers. Ever. Human and human? Nah, we're not going to do that. Chimp and chimp? Nah, not going to do that. Just bonobo and chimp. He doesn't want to do anything else because the method is bunk. And I think that's truly why he's doing this little coal hop dance here. I wanted to show you guys as well really quickly when I can here, just so you know that I tried to run it with the number of threads the way that Tompkins, just to head it off at the pass, and my computer gives me an error for this because my computer can't handle it. So that's why we're not using number of threads 6. We're doing it in a way that my computer can actually handle it. It like automates with the number of threads that I actually have access to, or that I can actually utilize. Okay. Oh yeah, people are talking in the side chat that it's misogynistic. This girl, You guys, I'm a woman on the internet and I'm a woman in academia. I know a little something about. I've been called girl more than once. I don't really care. Like he can be misogynistic if he wants. He can like bust my chops for being a woman and having a woman capabilities all day long. But none of that matters if he can't debunk my work, and he can't, or else he would. Okay, back at it.
I think a good example is, and I didn't watch this debate, but James Tour, who was kind of an origin of life specialist, recently did a debate, and the guy he debated, just was completely obnoxious and threw out all these ad hominem attacks and this and that, and it was just a circus. And so those are the people that are out there. It doesn't matter what you do, what kind of data you produce, they're going to come up with some sort of a diversion. And then, like you said, that's the great comment. Yeah, they all feed off of each other, and before you know it, they're creating all this nonsense out on the internet.
Just show why it's wrong. Just tell us why it's wrong, though, Jeff. That's all you have to do. And instead, he sits and whinges in the corner, and they're bullying me. They're attacking me. You don't see me doing this. I test your methods because I want to get to the bottom of it. And the bottom of it is that you are wrong.
And I can only I've only got so many hours in the day, so
Right,
yeah.
Right. I do have a question though. If your methodology was applied correctly, using the same system, the same tools and resources, if you did it yourself and just kind of apply it on, on same kind animals versus if you apply it like if they're exact copies of each other, would you get? Do you believe you would get accurate numbers regarding that? And two, would you actually do that? I mean, that sounds like a fun project. It sounds like something that would be a good way to actually see just how you compare those things. I don't know if do you think that other kinds of comparisons have been tried?
No, he's not going to run it. And yeah, as everybody in the side chat said, I didn't even catch it. I can't believe my brain is like so fried from so much Tompkins inundation. I didn't even check it. Origin of life specialist, James Tour. I don't know about that, Jeff. I don't know about that.
Very picked.
The thing is that when we talk about the human genome, it is the most heavily funded genome project in the world, because we want to get to the bottom of why do we have all these health problems and diseases and things. So they have actually sequenced human genomes all over the globe ad nauseum. And so I don't know what these people are doing, but the fact of the matter is that based on single base variations, we call them single nucleotide polymorphisms, or SNPs, and you can pull up any genome browser and all the SNPs are listed in there for the human genome. But if we just include the SNPs, or these single base differences, any two human genomes can be about 99% similar. Actually, I think the figure is 99-point-something% similar. But if you include the structural variance in between humans, any two human genomes can be 4 to 4.5% different from each other.
Okay, all right. So this is meant to be the fallback, the rescuing device, if you will, for what Jeff thinks that that Apologetics 101 is asking here, which is how come you're going to get a greater than 99% similar or less than 99% similarity between two people groups? And Jeff is like, haha, well, with single nucleotide polymorphisms or with structural variants, you can get up to 4% difference. Okay, first of all, I don't think you can get 4% difference in humans. Like I don't, because there's going to be overlap with some of the structural variants. Like I don't think you're going to ever get a 4% difference between any two humans. I don't believe that. Nothing I have read on the topic has ever suggested that you can get an actual 4% genomic difference between two humans. But that being said, none of that matters because your methods can't return a 100% identity for one person to themselves. We're not talking about two different humans with structural variants. We're talking about one individual with structural variants that they have to themselves, and you can't return 100% similarity. That's the problem.
If you include what's called structural variants, and which they are actually now doing, because the DNA sequencing technology with the PacBio sequencing, the ion torrent, and all these new technologies are actually able to sequence through the structural variants in the human genome. And so if you include those, any two human genomes can be up to 4 to 4.5% different from each other. Well, if we have 4.5% difference from each other potentially, how are we only 2% different from a chimp? It's nonsense. And these people that claim, oh, I discovered this by comparing the same genome. I mean, you know what? The entire global human genomics community has already done it. You can just go to the literature and see what they did.
Yeah. So again, he doesn't understand what's being asked of him here. Just putting that out there.
All right.
So anyways, and they found pretty much the same situation among populations of chimps that they've sequenced as well. So I don't know what these people are doing, but yeah, as far as comparing one human genome to another, it's been done ad nauseum all over the world in the Human Genome Project, which has been going on since early 1990s.
Right. I do have Erica from Gutsick was actually in the live chat when you did your.
Oh my God. Does he? Okay, I think I understand what's going on here, actually. So I think what Jeff is saying is he's like, why would I ever compare two human genomes using my methods when everybody else has already done it? What kind of dissonance is going on in your head at that point, right? Like to control your results for human and chimp, that's why you need a control. But he doesn't want to control, right? Like that's what this ultimately comes down to.
Your interview with Christopher Roop, or what was his name?
Yeah.
Christopher Roop? Yeah. That was her. She did give me a question to ask you, though. I quoted it exactly. This is the question that she had verbatim. If your genetic work can differentiate between kinds and separate humans from chimps, why are so many pairs that you accept as related less similar?
Okay, you guys. This is the question Apologetics 101 asked me in the chat. He was like, give me a question, ask Jeff. And that's exactly what I asked, right? A little bit paraphrased from what he just said. If your work is meant to differentiate kinds and shows, like your genetic work, Jeff, is meant to differentiate kinds, and your genetic work is showing as humans and chimps is hashing out as separate, right? My question for him is, how does that square with the fact that you can use his methods and get animals that he thinks are the same kind to be similar, like more different than the human and chimp, if that makes sense? So let's hear him say it again.
Chat when you did your interview with Christopher Roop, or what was his name?
Yeah.
Christopher Roop? Yeah. That was her. She did give me a question to ask you, though. I quoted it exactly. This is the question that she had verbatim. If your genetic work can differentiate between kinds and separate humans from chimps, why are so many pairs that you accept as related less similar? Okay,
that's a straightforward question, and I'm going to pause for a second here and allow. I want to ask everybody in the chat, right? Like people in the chat, was that a clear question to you? Do you think like was that clear what was being asked there with all of the context of everything going on? Was that clear what was being asked? I want to know. I need to know. Because moving forward, Jeff is going to say something quite silly. Okay. I'm getting yes, straightforward, yes, yup. Okay. Okay. The question is clear. All right. Excellent. Clear enough. So let's let him continue. With that in mind, let him continue. I just clicked on the wrong thing. I'm getting fired.
I'm not sure I understand the question.
See, if your genetic work can differentiate between kinds and separate humans from chimps, why are so many pairs you accept as related less similar? I don't understand the question either. Now think about it.
Apologetics 101. You had me in a chat. You could have asked me to clarify. You could have emailed me. I told you email me if you needed clarification because I knew you were going to ask this to Jeff. You don't even know what I was asking. Like this question is like one-to-one related to the work that I did in that video. But you don't know what the question is. And Jeff doesn't know either, or at least he's pretending not to know. I thought it was straightforward, but
I mean,
I don't know how to answer that.
I don't either. This is verbatim. This is what she put in the chat for me to ask. I don't know. I think it might be relating to maybe a fundamental misunderstanding that we think all kinds are exactly alike, or but we younger species actually accept the possibility of biodiversity among kinds. So, like, what do you do with that, right? Like, I don't know how you could watch my entire Tompkins video and come away with that as the concept. No, the problem is genetics cannot differentiate kinds of animals, as the creationists put it, in such a way that can group animals that they accept are related, like lions and tigers, African and Asian elephants, black bears and polar bears, that can lump all of those guys together genomically speaking, and also parse humans and chimps as apart from each other. Humans and chimps are 98.8% similar coding base pairs, up to about 99.1% for coding base pairs, but at least 96 for the full genomic comparison. That's greater than tigers are to lions. It's greater than African and Asian elephants are to one another, right? So
presumably, genetics should be able to, if kinds are a real thing, distinguish between kinds in such a way that is going to apply to humans versus other animals as well, but it doesn't. That's the problem. And the problem gets even worse with Jeff's methods, because using his methods, some of these pairs that are like definitively absolutely related, like Bos indicus and Bos taurus, two different kinds of cow breeds, are showing up as less similar than humans and chimps. That is not like, that's not reality, right? The Bos indicus and Bos taurus are more similar than humans are to chimps, but they don't show up that way in Tompkins's methods. This is the issue. So like, how do you deal with that concept broadly speaking genetically, and how do you deal with it within your own methods? Either of those I would have accepted an answer to, but neither of them are what we actually got.
Right. And you know what? Every kind of creature is different. You know, there's more variation within some kinds than there are other kinds. You know, nothing is so. You know, some creatures there's very little genetic variation between them. Other types of creatures there's a lot, but yet they don't interbreed with other fundamentally different kinds of creatures, and they don't evolve into them. Right. And so,
no matter what, though, two creatures that are within the same kind should be more genetically similar than two creatures that are in different kinds. No, if that's not the case, then you guys, genetics can't distinguish kinds, period. It's useless with kinds.
Oh,
and by the way, that makes kinds arbitrary nonsense, as we always knew that they were.
Life is, life across the board is so amazing. It just really is. Right. All these genomes and genomic and cellular systems are so, mind-bogglingly complex and amazing. Yeah. But yet, the critics are basically all nitpickers. And so they want to find some little anomaly and ignore the 99.9% of the data that screams there's a creator.
It's not an anomaly. It's a fundamental flaw. Like, I don't know how much more straightforward I can be.
And that's their mindset. Let's nitpick this little thing here and see if we can find something wrong with it, but it has no bearing on the broad picture of things. And so that's really common. I see that across the board. It doesn't matter what I'm talking about. They want to find some little nitpicky little thing and focus on that, but really ignore the bulk of the evidence. And so, like I say, it comes down to your presuppositions. Are you just dead set? There's no creator. We all evolved from pond scum. If that's your mindset, then you'll say anything or do anything.
Yeah, I mean, this is just dumb. Like, this is really uncharitable and dumb. Like, if I don't behave this way with other people, I don't assume that they automatically are unreachable. You have to prove that to me. And Jeff has proven to me that he is, in fact, probably unreachable. Presuppositionalists are kind of a waste of time to argue with, but I do think it's worth taking on some of their challenges, because presuppositionalists do a lot of the creationist science, quote unquote, legwork, because they see the fact that they presuppose God as like Abrahamic young earth creationist style God as true, as kind of like a motivator, right? And then they go out and try to prove that point, which is the opposite of how science is run. But anyways, they kind of take that stance. And so they do a lot of the legwork with this. And it's worth taking the time to prove presuppositionalists wrong on certain points, or to show that their points don't hold very much water, or any water at all, about as much water as like a pasta strainer, because it can be useful to other people who maybe aren't so rigid in their interpretation of the Bible or in their understanding of science and what science is capable of.
And you'll ignore the bulk of the evidence. The Bible says that people are without excuse. There's so much evidence out there. Romans 1:20. There's so much evidence out there that basically people are without excuse. And you want to come up with all these little nitpicky, foolish arguments. You're doing it to your own destruction, eternal destruction.
Foolish arguments by someone who can't weight their data.
I like that.
You think that they would target the whole accusation about cherry picking, because that's the main point of all this, that you're cherry picking the data. This method eliminates that cherry picking. Right. And you think that they would sit there and just say, no, we're not cherry picking. But instead of doing that, they challenge the method instead of the point of the method, instead of point of the whole argument.
Okay, so my understanding is that the most recent work, as far as comparative genomics goes, is like exactly meeting Tompkins's expectations. It's using the most up-to-date version of the human genome, which is complete, the most up-to-date version of the chimpanzee genome, which is almost complete, and comparing them, like both of which were independently assembled, and comparing them whole genome, right? And the number is still 96 for full genome, 95 to 96 for whole genome, 98.8 plus for protein coding regions. Like, I don't know what more you want. I mean, I know what he wants. He wants people to agree with him, but like, He also simultaneously won't publish in like a conventional journal, not this work. His other stuff he will, but not this work. Kind of makes you think he knows where the flaws lie, doesn't it?
Yeah, and it's, you know, they like to come up with these little esoteric arguments that basically people don't understand anyways, and much less probably them.
The irony of that statement, right?
So yeah, it's yeah.
Right. All right. If you could you summarize and come to the conclusion to your overall case, and then what, you know, based on your conclusion of your findings and so forth, and give us your final thoughts.
Sure. So like I said, you know, when I got into this whole issue of whether humans, and guy there's myself. So anyway, right human to stuff rhythms they were using.
He doesn't really say anything new here. I like I said, I already watched this, so I don't really think there's anything here. I don't think there's anything else here worth covering, to be perfectly honest. Oh, actually, there is one thing.
On the bugs article, you said that bugs agreed with 84% in a blog. Supposedly, he wrote a peer review paper later, changing the number to 96%.
Okay, so here's the background here. Tomkins utilizes the work of Richard Buggs, like an actual legitimate scientist. He does fine work, whatever, to kind of like bolster his own 84%. Because Richard Buggs said he's put forward the 84% number as this similarity for humans and chimps, but he does it on the basis of something else. He does it on the basis of like how you handle indels. So like I said earlier, you got weighted, unweighted, that's a problem. Gapped, ungapped, that's a problem. And then how do you handle indels? So Buggs is making his arguments on a completely different foundation as compared to Jeff. And when Buggs was making these arguments, he was doing so, again, on his blog, on the BioLogos forum, etc. So he doesn't really say very much about it. And then he goes on in like 2020, publishes with a student a paper on like using, it's like what is it? It's like an imaging, it's like looking at the pictorial content, something like that on Chromosome. Let me just find it. Hold on. Richard, I think it's in Frontiers. Frontiers? I think it's in Frontiers. Chromosomes, maybe I put that. Maybe that'll help. Chromosomal. Okay, here we go. It is called Fluent DNA, Nucleotide Visualization. Why did I keep saying pictorial? Visualization, that makes a lot more sense. Okay, so I'm not going to switch it over because it's not really worth it, but it's called Fluent DNA, Nucleotide Visualization of Whole Genomes, Annotations, and Alignments. And it's by Richard Buggs and Seaman, his student. And they come up with a 96% for the similarity between humans and chimps. So the thing that Tompkins doesn't like here is that Tompkins is like, oh, well, maybe Richard Buggs did publish with his student on a 96% similarity between humans and chimps. But earlier he published in a blog that there was an 84% similarity. So who will you believe? And it's like, I'm going to believe the published paper, because like Buggs could have just published on the 84%. And like he probably, I find it hard to believe that Richard Buggs published in a blog the 84% number, then published in an actual conventional science magazine or journal, excuse me, the 96 number, but he actually just still holds to the 84 number, right? I don't really believe that. I find that hard to believe, especially because the evidence that Tompkins presents is that someone told him, sorry, I got a dog. I got several dogs. Someone told him that Buggs said that, right? Someone else told him that Buggs said that. So let's hear him make that case.
Why does Buggs change it to 96% later? What was his reasons for that?
Well, the guy that wrote the paper was Joshua Siemens, and he did a PhD with Buggs. And Joshua Siemens was creating DNA visualization software. So the software wasn't necessarily intended to do these really genome-wide alignment comparisons.
I mean, you say that, but the title is Fluent DNA, Nucleotide Visualization of Whole Genomes, Annotations and Alignments. So I don't know that you can make that case, Jeff.
Necessarily. In other words, the 96% talked about just the regions that were highly similar, that did line up. And someone sent me an email from Richard Buggs. It was very recent. I think it was within the past year, which was after he published that paper, where he claimed in the email that the 84% still stood, that it was still valid. So Joshua Siemens' genome visualization software was.
Okay, there you have it, you guys. The source is someone told me so. Okay. If you say so, Jeff, if you say so.
Was not a sophisticated, BLAST-N or BLAST-X algorithm. It was just visualization software with a very crude brute force algorithm.
Awesome. Okay. And of course,
And I actually talked to Joshua Siemens at some meetings after that was done, and yeah, it. Anyways,
right. And of course,
I wouldn't put a whole lot of stock, in that.
right.
Dude, the irony here. Jeff wouldn't put a lot of stock in a published paper that Bugs was on, but him hearing from somebody else that Jeff said in an email the 84% still stands, that he puts a lot of stock in. Cool, Jeff. Really cool. Okay, you guys. I'm not sure there's anything else here. I think he just does another witnessing thing and then kind of finishes. I think that's pretty much it. I think that's the video. That was Jeffrey Tompkins' response, I guess, to my video that I made. And to sum it up, gosh, where do we even go with this? Okay, to sum it up, Jeffrey Tompkins thinks that my experiments were not correct because I didn't use a big enough computer. And therefore he doubts that I actually ran his methods. He thinks my methods are incorrect because weighting actually doesn't matter. No, he did not elaborate on that. He thinks that he's still correct because Richard Buggs still agrees with him, even though Richard Buggs wrote a paper that he was on, or at least he was a second author on a paper with his student where the number is 96% between humans and chimps. And no, he does not elaborate on any of the actual work that I did. He doesn't comment on any of my actual tests. He doesn't run any of my actual tests. And his conclusion is kind of just like, nuh-uh, I'm good. My work is still good. So that's pretty much what Jeff had to say. Very disappointing, actually. The only good thing really about this interview is I feel like I learned a little bit more about Jeff. And I learned that he's kind of a jerk. He's kind of an asshole. And like Jeff can get mad that I'm being crass here, but like I just don't care. I don't care what Jeff has to say. I don't think he's a very good geneticist. I don't think that he's a very kind person or a very charitable person. And I frankly am very much looking forward to another decade of whinging and obfuscation from him. I hope moving forward that young Earth creationists and other types of creationists will continue to abandon Jeff's numbers as they've been doing. Evolution News, as I included in my previous video, they don't even like name drop Jeff, which I think is absolutely hysterical. They name drop plenty of other people. Anyways, yeah, humans and chimpanzees are 96% similar to one another. Whole genome comparison, 98.8 plus for just the protein coding regions. And we are more closely related to one another than either of us is to anything else, and more closely related to one another than lions are to tigers, or to cats, or to lynx, or African and Asian elephants are to one another. And using Jeff's methods, humans aren't even 100% similar to one another, like to themselves, I guess I should say. I don't anticipate we're going to get any more responses from Jeffrey Tompkins on that topic, because if he were to actually go out and test what I did, well, he's going to come back with some answers that he doesn't like. And I think actually, as I've mentioned before in that clip I mentioned, I think he's probably done that already and has just decided to keep a wide berth from running any kind of analysis that might systematically destroy his entire worldview. Anyways, you guys, if you like what I do on this channel, please consider liking, commenting, and subscribing. It's a great free way to support me and help make sure that I can continue to do this. And if you want to support me in other ways, you can become a patron member. And when you're a patron member, your name rolls at the end of the videos in front of one of my animations. And you get early access to videos, which all the patrons right now have access to every video that I'm releasing while I'm in Kenya. So that's fun. And my gentle and, of course, very modern apes, please do take care of yourselves, and I'll see you in the next one.